Hello, and welcome back to PPMD's 26th Annual Conference. I'm Eric Camino. I am PPMD's Director of Research and Clinical Innovation.
I'm Ryan Fisher, Senior Vice President of Community Engagement.
We're so glad to have everyone here with us again tonight. For those of you who missed last night's session or Wednesday night's session, you can go and view those online now. They are up on our hub, so if you go in, you can go check out all that great content that we had yesterday. We had a number of great sessions. Our first one last night was talking about resiliency, which I thought was great, Ryan.
Yeah, it was a great way to start the day. I think it's something we all need right about now. The topics focused on compassion fatigue and caregiver burnout, things that we may not recognize are happening in our community. We had a great panel of PPMD Connect coordinators. We want to thank them all for being on from different states. And we had Dr. Natalie Truba and Dr. Kozar from Nationwide Children's to talk about what they see with their families and how they give them guidance and tools to deal with things like coping and burnout, real issues in our community. And I took some notes, just some of the things that they were saying during the talk really resonated with even myself, focusing on the positives and talking to your family about, what went well that day and why, and really focusing on the why. It was a great take-home message, and finding your us, the people who support and love you, your village essentially, your support system. These are people within our internal Duchenne community, but also your external community within your own town, as well as your family members. So, and I love this. The past does not define me; rather, it trains me. So, be sure to check that out. I think everyone could benefit from watching it. It was a really great session. We also had our in the pipeline series. We tackled restoring dystrophin, didn't we? Yeah, no, it was another great panel on our In the Pipeline series. So we got to hear from a number of different sponsors looking at trying to restore dystrophin through either a stop codon read-through or exon skipping therapies. And again, we hear about kind of this exciting time where we had a number of therapies that are approved or on that pathway to approval, as well as some companies that are in a little earlier stages. But we got to hear about that, some of the exciting new technology that's coming along with those exon skipping therapies. So it was just an exciting time to talk about those. Yeah, and as we were listening to the talks, one of the things I think we all recognize is how painfully slow some of this drug development is, and the fact that there are exons in the pipeline and coming soon, and we're working on these, and we do appreciate the companies for all they're doing. But we also know it can just be painfully slow to be waiting for an exon to get past the preclinical stage, and that, in general, drug development takes a really long time. It's why, as an organization, we're focusing on ways to speed the development of these therapies, and whether that's creating a more sustainable path or talking to the FDA, working on legislation that incorporates that makes the FDA incorporate the voice of the patient into the process. These are all pieces that fit together that we need to really speed the development of drug development. So, I think it was a good session, but hopefully more to come, and we'll continue to bring those companies via webinars to the community for conversation. So, some housekeeping. We want to remind everyone, if this is your first night joining us, to bookmark the hub. This is your go-to place, of course, for all the information about conference. This is where you're going to find pre-recorded materials. This is where you sign up for Zoom socials. It's where you need to find the most up-to-date information about conference. We do want to take a moment to thank our sponsors. Tonight, we're giving a shout-out to our gold sponsors, Diasha Sanko and Fibrogen and Roche Genentech. Thank you very much. And then silver sponsors, Astellas, Audentes, Edgewise, and Vertex. So we couldn't do this conference without any of you, so we really do appreciate it. So with that, Eric, what is up today?
So for today, we have another really packed day. We're going to kick it off with our In the Pipeline series, and we're going to be talking about downstream targets. So, we talked about some of the exon skipping. Those are mutation specific. We're going to be discussing some of those therapies that can be, used broadly within the Duchenne population, as well as the Becker population. We're going to be taking a look at a number of different care areas for Duchenne. So we're going to tackle endocrinology, and we're also going to take a look at acute pulmonary management. Additionally, we have our gene therapy session tonight, and Dr. Tim Kripe from Nationwide Children's Hospital is going to join us to give us a little introduction to that session before hearing from some of those sponsors. For our teens and adults, we have a private Zoom discussion tonight so they can discuss about living with Duchenne, and so that's for teens of 14 and up and our young adults. And then we have our Zoom socials. So for our moms, dads, grandparents, and siblings, you can all meet up together and have a nice little social after we wrap conference tonight, give you a chance to maybe discuss some of your experiences with other people who may be going through similar life experiences as you are. And so with that, we are going to get started with our first in the pipeline series for the evening and talk about downstream targets. And so when we talk about downstream targets, what we're really discussing are all of those kind of effects that we see because of that lack of the dystrophin protein. So as we kind of discussed on Wednesday night with inflammation, that without the dystrophin protein, we can see inflammation, we can see the replacement of muscle tissue with fatty tissue and scar tissue, we can have calcium imbalance, cellular energy depletion, and all of these things can contribute to the disease progression that we see. And they don't just affect the skeletal muscle. We also see this impacting the cardiac, so the heart being a muscle too. So the strategies that we are going to talk about here in this session are going to focus on providing benefits by tackling kind of these downstream areas. So we're going to hear about some therapies that look at improving the function of the mitochondria. And then we're also going to hear about a therapy that's looking at providing some benefit to the heart. And I think it's really important that we, think about these therapies because although restoring dystrophin is very important and something that we look forward to, we know that exon skipping and even gene therapy aren't going to restore 100% of the dystrophin in the muscles. And so we still have these areas that are going to need to be addressed. We're going to have to take these therapies in combination with other therapies. So it's really important to continue to see the development of these different types of potential therapeutics. So I'm going to be joined tonight to discuss these by Dr. Gerard Merrick from Astellas Pharma, Dr. Shabir Hasham from Santhera Pharmaceuticals, Dr. Larry Markham, he's an MD and he's representing Cumberland Pharmaceuticals, and Dr. Ranzi Samarante from Aperium Bio. And so, as a reminder, if you have any questions for any of our panelists, please submit them in the comment box below and just indicate what sponsor you are addressing that question to. So Dr. Merrick, I think we are going to get started with you.
Okay, thank you very much. I'm Gerard Merrick from Astellas Pharma, and if we could go to my next slide, then I'm going to be talking about ASP0367, which was formerly known as MA0211. And this drug, the target that it's acting on is a protein called PPAR delta modulator inside muscle cells. And we potential benefit that 0367 may act inside the muscle cells to actually increase the mitochondria's ability to produce energy. And mitochondria is sort of the powerhouse inside each cell. We're talking about we've completed phase 1 studies in healthy volunteers, and this next upcoming study is a phase 1B study that will be opening soon. We hope to have it opening up actually approximately in November for screening. We'll be looking for boys ages 8 to 16 to volunteer for this study. Some of the endpoints we'll be looking at is a novel one called a 6-minute cycling test, which is simply is an arm cycling test. Some of the secondary measurements include pull, 2-minute walk test, respiratory function, and accelerometer, as well as other biomarkers. And we've been assigned to orphan drug designation from the FDA. And if the safety and preliminary efficacy signal in the upcoming phase 1B do work out, then we will be investigating efficacy and safety in phase 2 and phase 3 trials in consultation with the FDA. Next slide, please. So, as I mentioned, A0367 is being investigated to determine whether it can turn on PPAR delta pathway. And this PPAR delta pathway regulates the mitochondria on different genes in the cell. And when this pathway is on, the mitochondria are able to use fatty acids more efficiently, and more mitochondria are made. The mitochondria are working better, more efficiently. So this use of more fatty acids for energy results in increased energy production inside the cell. Next slide, please. So this phase 1B study is going to be a randomized, double-blind, and also placebo-controlled. The primary purpose is really to evaluate the safety and tolerability of ASP0367. We'll also be looking at how the body processes the drug, as well as I mentioned, how the drug affects function, looking at this arm cycling test. And trial sites we plan on opening in November of this year. And to find a participating site near you, you can visit the clinicaltrials.gov and search NCT 041-848-882. Or you can contact Astellas Pharma at the number that provided here, 800-888-7704. Next slide, please. So as I mentioned, for the study schedule, we have a 4-week screening period. This is followed by 2 weeks on a low dose of the drug, and then 10 weeks on a higher dose of the drug. And so this happens both for those subjects on active ASP0367, and about half as many subjects will also be on placebo during this 12-week period. After this double-blind placebo-controlled part, all subjects can go on to receive active ASP0367 for additional 12 weeks, and there'll be a 4-week follow-up. We have approximately 15 visits throughout this study, and what we just put in an amendment to our protocol, which has made a change where we've we've made about a little bit over half of these visits into home visits, just to minimize having to go into the clinical site, just as has been true for many of you in terms of normal clinical care as well. Next slide, please. So who can participate in this study? It will be boys 16 years old, having obviously a diagnosis of Duchenne muscular dystrophy, also taking stable doses of corticosteroids and any cardiac therapy that you're on, and having some trouble walking quickly as defined by the 10-meter walk test during your first screening visit. But you need to be able to move your arms up at least to the shoulder height. Exclusion criteria include acute illness or serious infection or other problems suggesting you're not healthy enough to participate in the study. Also having heart problems, like your heart not pumping well, where the clinical term is an ejection fraction, not being good enough, or long QT interval, meaning the heart doesn't send electrical signals well enough, or having high levels of cardiac troponin on a lab test, suggesting there may be other issues with heart cells. And also exclusion if you use certain types of drugs within the last 4 weeks of the study, or having kidney or liver problems, or currently receiving an investigational drug, or if you've ever taken an exon skipping or gene therapy drug. Next slide, please. So then what happens at the study visits? We'll be looking at your general health by Dr. Checking at study visits and blood and urine tests. We'll be looking at how your heart is doing with ECG echocardiogram, doing breathing tests, pulmonary function tests, how well you move your body, such as in the arm cycling test, performance of the upper limb test, 2-minute walk test, and other tests at home, including video tests and activity monitor. We'll also be looking at how your body uses the drug in terms of blood and urine tests, and then how you feel and think, a range of tests, including surveys, diaries, and interviews, and also looking at, like, for example, digit muscle tests and other things. So I'd like to thank you for listening to our presentation, and I'd like to thank PPMD for the ability to present our study as well. Thank you very much.
Thank you, Dr. Merrick. That was wonderful. One follow-up question I had for you around that exclusion criteria. I saw that was mentioned mental health problems or behavioral problems, and certainly, as you understand, because of the absence of dystrophin in the brain, we certainly have a number of patients impacted by psychosocial issues, diagnosed with different conditions. Can you define what is meant by mental health problems or behavioral problems for that exclusion criteria?
Well, that's really one where we're leaving that to the discretion of the PI, the investigator. So, I mean, they just have to make the judgment whether or not the boy can actually engage with the staff and participate in the study and follow directions. So, basically, it's can they follow directions for the study procedures. That's what it really implies.
Okay. Oh, thank you so much for that clarification. Wonderful. Okay, so we're going to move on to our next speaker. Dr. Shabir Hasham from Santhera is on with us, who's going to talk about what's happening with Santhera and their trials. And Dr. Hasham, are you there?
Thank you very much. My name is Shabir Hasham, Santhera Pharmaceuticals, and I'm delighted to present to you an update of our late-stage clinical development product, idebenone. And I just wanted to thank everyone for the opportunity to share the information with you. If I could have the next slide, please. So Santhera has been focused on investigating the advanced phases of Duchenne. Predominantly, we have an emphasis on trying to demonstrate the benefits of preserving lung function and the long-term benefits of doing so. We also have a product that targets mitochondria, and in our case, the product encourages electron shuttling across the respiratory chain, reduces oxidative stress, and increases ATP, and by doing such, allows respiratory muscle cells to function better and in some ways helps to prevent further muscle death. We have a late-stage development program. Data from our phase 3 Delos study was published in 2015, and this was in a population 10 to 18 years old. And these patients were in respiratory function decline, as we defined as having respiratory function of less than 80% predicted. Treatment with idebenone showed that we were able to statistically reduce the loss of respiratory function as measured by peak expiratory flow, but we also showed consistent benefit across forced vital capacity and forced expiratory volume. Importantly, we were also able to show by maintaining lung function and reducing the loss where possible, we actually reduced the risk of having infections, bronchopulmonary infections, and also the risk of hospitalization due to respiratory complications. Next slide, please. Whilst this is an important first step in building the knowledge about the potential long-term benefits of maintaining lung function, we've also been working quite extensively with academic institutions and doing modeling in natural history to try to understand what benefits could be brought in terms of long-term delays to clinically relevant milestones. I'm going to use this graph just as an illustrative example to try to bring across our key learnings. We examined natural history data in two populations, one in which respiratory function was declining faster, here shown in red, and compared it to one in which respiratory function was declining slower, here shown in blue. And what we were able to show was that those who were declining faster crossed clinically relevant thresholds of lung function that are known to predict an increased risk of infections and also the need for non-invasive invasive ventilation. What we were able to do in the modeling was to show by slowing down the rate of lung function loss, that population also crossed the clinically relevant thresholds, but later, and led to a delay in the need to start assisted ventilation. And this forms the basis of our continued work going forward in terms of trying to build the evidence base about a long-term benefit in preserving lung function. Next slide, please. We recently announced the completion of recruitment of our large phase 3 SIDROS study in planned 266 patients. These were in patients 10 to 18 years of age, again, in respiratory function decline, having an FVC of less than 80% predicted, as opposed to the DELOS study, which was investigated in patients who were not taking steroids. This study is looking at the combination of idebenone with glucocorticoids. We had an opportunity as part of the protocol to do a sample size re-estimation, and this really allows us to make sure that we have a sufficient number of participants within the study. The study was originally designed to have a 90% power, and that's the ability to detect a true treatment effect of the drug. When we did the protocol sample size re-estimation, we were able to look at blinded data. And what we saw that the respiratory function test, the FVC, the spirometry, and the peak expiratory flow tests performed much better than we had originally assumed in the study design. And so we went from a power of 90 to more than 99%, meaning that the study is very well powered to detect a true treatment difference. What we also were able to do was to determine that almost half of patients who were currently in the SIDROS study had completed the 18-month observation period. And therefore, we're in a position now to conclude that we have enough data to conduct an interim analysis. Now, what that means is that if we are able to show compelling efficacy in this interim analysis, we're able to actually stop the study almost a year, if not a little bit more than a year ahead of schedule, and then start to have negotiations with the FDA for an NDA application somewhere in Q2 in 2021. Next slide, please. I just wanted to share some insights in the impact of COVID, and I'm sure many of my colleagues here have also been having to think about ways that we can manage COVID and the risk it has to the study. The team at Santhera, in collaboration with patient groups, but also with the community on a whole, have done a tremendous job in trying to participate in clinical trials. We were able to have strategies to deliver drug to home, remote monitoring, and also the possibility to try to extend visit windows. And I'm pleased to say that the impact of COVID on the SIDROS study has been quite manageable, if not minimal. Thank you. Next slide. So in summary, Santhera continues to expand the data and knowledge that we have in terms of preserving lung function, delaying the loss over time, and the benefit that that may bring in the long term in terms of delay to needing assisted ventilation. Data from the pivotal phase 3 DELOS study had previously demonstrated that Vuldisa has been able to slow respiratory function decline, reduce the risk of infections and hospitalizations, and we hope that the phase 3 SIDROS study will continue to build on that knowledge base in a much larger group, but importantly in combination with glucocorticoids. Analysis done during the sample size re-estimation has given us the opportunity to stop this study early if we're able to show compelling efficacy in the interim analysis. And we hope that we'll be able to communicate to the community the outcomes of that by Q4 this year, which means that we may potentially be able to accelerate time to market by more than a year. And I'd like to thank everybody who's continued to participate in the study. I know it's been a very, very difficult time. And we just want to say thank you on behalf of the company and investigators and sites for the continued commitment that will help potentially to bring this study to an early closure with an outcome that could bring a therapy to market a year in advance. With that, it's just to say thank you to PPMD for the opportunity to share this information with you. Thank you, Ryan.
Thank you, Shabir. I really, really appreciate that presentation. That was really helpful. For some in the community who may not have been following for many years the work you all have been doing, could you sort of talk through your thoughts about why, with the original phase 3 meeting an endpoint, which was, I believe, the first trial to do that in Duchenne, why the FDA may have been reluctant to move forward with that and explain to them sort of what happened after that and the path you took?
Sure. Thank you for the question. It's a very relevant one. So with DELOS, we built on the experience of the previous phase 2 Delphi study in which we had been able to show a treatment effect originally in peak expiratory flow. And we used that as the basis to design the phase 3 study. And at the time, of course, FVC as measure was the preferred measure for the FDA. But in discussions with the FDA, we decided to proceed with the Delos study. But since then, we've been trying to establish the evidence base to show that actually PEF as a measure, clinical measure, is interchangeable with FVC. And that's the current focus, certainly within Europe and the regulatory strategy and submission there. We had a very clear understanding from the FDA that Sideros was the study that they would be interested in. And that's why we're so excited that we have the potential now to bring it to an early termination. Of course, it uses FVC, which is the preferred measure for the FDA, as well as using peak expiratory flow. So both studies are actually supportive of each other. And so we will have two studies to be able to show in a phase 3, obviously randomized controlled study, the benefit of Puldisa in terms of trying to delay respiratory function loss.
Thank you. That was extremely helpful. And I just want to acknowledge you all have done a lot of work to advance drug development for the non-ambulatory population. And we truly appreciate that. And we know that done a lot around endpoints that are needed to be advanced for this population. So we are grateful for that. Okay, so we are going to move on to our next speaker from Cumberland Pharmaceuticals, Dr. Larry Markham. Are you there? And please take it away.
Yes, I am here. So I'm Larry Markham. I'm a pediatric cardiologist living in Indianapolis at Riley Hospital for Children. And I've been working with Cumberland for the last couple of years. So next slide. I have no direct financial conflicts. Next slide. So we've been working on looking at efitelban in the Duchenne individuals. So efitelban blocks the thromboxane prostanoid receptor, which is expressed on many muscle cells, but also platelets and immune cells, and contributes to fibrosis in the heart and lungs. And so treatment with efitelban has the potential to prevent fibrosis and inflammation. It's an oral medication, which is administered once daily, and safety is well established in other disease indications. Next slide. If you would like some more background on the basic science information, you can check out poster 14, which has been uploaded. And this is work that I and a team collaborated on with investigators at Vanderbilt. But this is a cartoon of the pathway. And you can see there at the top, aspirin is one drug that also interacts in this pathway. But efitelban blocks all of the cell surface receptors, and as such, blocks some of the profibrotic signaling pathways. Next slide. So working with PPMD, Cumberland, Fight DMD, colleagues at Vanderbilt, this has been a process that has been going on just over 3 years. And last fall, this was awarded the first DMD trial, awarded an FDA orphan grant to study this drug in Duchenne, and was also sponsored by Fight DMD, which is a foundation based in Nashville. Next slide. This is probably the most important slide, is the study design. So this is a phase 2 with the objective to evaluate safety, efficacy, and pharmacokinetics. 2/3 of the subjects will be randomized to efitelban, 1/3 will receive placebo. The treatment duration will be 12 months, and there will be 3 visits. At visit 1, will be randomized individuals to low dose or high dose. Some of the criticism centered around, if you take individuals who all have normal heart function, how will you tell if it works? And so we have 2 groups of individuals getting treated, those with mild cardiac involvement or no cardiac involvement, and those who have a little bit more moderate cardiac involvement, to see if we can rescue those who already have significant cardiac impairment. Next slide. There are some key eligibility and study features. This is an option for all Duchenne boys greater than 7 who can tolerate a cardiac MRI, regardless of mutation type, ambulatory status, or steroid use. There is no upper age limit, and even those who are on exon skipping therapies will have possibility. And because it includes patients with all ambulatory categories, there is no walk test and no biopsy. Next slide. This is a map of the sites. Currently, Children's National in DC is up and running. As Shabir said, we've all been impacted by COVID and how we can get things going. Here at Riley, we're close. We're probably the second site that will be up in this list, and all we need to have is our site visit. And then there are several others that are coming on rather quickly now that we have returned to some research activities. If you do have an interest, there is the address and email for consideration of being involved in the study. And again, as everyone says, I know the conference has been significantly impacted. And so we just appreciate the opportunity to be involved with the community in this way. And I think the next slide is just questions and thank you. So you can click through those. Thank you.
Great. Thank you, Larry. Really good to see you virtually. Just one quick follow-up question before we move to the next speaker. I think families are probably wondering, are you envisioning this is going to be in combination with standard of care cardiac medications in the future, and that's sort of the plan for treatment?
Yeah, I think as we've looked at this and looked at things over time, I think we are starting to understand that scar tissue or fibrosis in the heart is a very early phenomenon, and a lot of the treatments that we do, ACE inhibition, beta blockers, and a eplerenone or spironolactone, they all target fibrosis, but in different ways. And so I think if we have another option to add to that collection of drugs, then I think it will be a good thing.
I definitely agree. I certainly agree. Combinations are needed, and we appreciate that. Okay, we will speak to you on the panel in just a bit. Our last speaker of this session is from Imperial Bio. Ronzi, you there? Wonderful.
Yes, I am.
Please take it.
Can you hear me? Great. Thanks very much. First slide, please. Thanks very much. I'm Ronzi Somaratne, Chief Medical Officer at Imperial Bio. As Eric so nicely outlined at the top of the presentation today, the lack of dystrophin leads to a number of downstream consequences that end up leading to fibrosis, fat deposition, and we see this as a vicious cycle. And the hypercontractility-induced injury in the skeletal muscle fibers leads to inflammation, loss of mitochondria, loss of mitochondrial efficiency, accumulation of reactive oxygen species, fibrosis, which leads to more damage, and this cycle continues. Next slide, please. So what we've learned, and our lead candidate is called EPM01. We believe that this compound shows potential benefit for patients with both Duchenne and Becker muscular dystrophies. And what we've learned through a couple of small pilot studies using EPM01 and an earlier, less potent compound is that there appear to be multiple potential benefits for these patients, such as improved energy balance in cells through enhanced production of mitochondria and enhanced mitochondrial function, a reduced inflammatory response in muscle, a reduced amount of oxidation injury, improved balance in the proteins regulating muscle regeneration and growth, including follistatin and myostatin. We've noted the increased production of nitric oxide. In the Becker study, we noted increased expression of key proteins such as dystrophin. We've seen encouraging data on cardiac function, including effects on brain natriuretic peptide and troponins, and we've also seen encouraging trends in exercise capacity. Next slide, please. So through the multitude of effects that we've noted, we believe that the EPM01 and this class of medication could improve all the various stages of the cascade of events occurring after loss of dystrophin and potentially lead to clinical benefits in patients. Next slide, please. So where are we currently? We have just started up a small study in patients with Becker muscular dystrophy. We're looking to enroll 20 patients between the ages of 16 up to the age of 60. We're primarily looking at the safety and tolerability of 3 doses in anticipation of the future trial. We're going to be following these patients up for a total of 1 year on drug. We'll be doing things like the timed muscle function test, which you're all accustomed to in this type of clinical trials, looking at key muscle proteins and other biomarkers. We'll be looking at cardiac assessments, including cardiac MRI via core lab and BNP. And we're really now moving the program towards studies in Duchenne muscular dystrophy. We're still trying to work out the clinical research phase, the target ages in Duchenne, and the endpoints that might be of importance in our studies. We're also working with regulatory agencies in defining the pathways for both the Becker and Duchenne programs. I should note that we've been granted orphan designation for Becker and Duchenne muscular dystrophy by the FDA. Next slide, please. So key takeaways, we believe that our compounds have a unique mechanism of action that's directly relevant to Duchenne muscular dystrophy. It seems to restore bioenergetics and muscle structure. We've now, through a variety of prior studies, have safety in over 100 patients, both adult and pediatric, across a multiple set of diseases. We have been encouraged by the types of biomarker movements we've seen in both Duchenne and Becker muscular dystrophy. We've seen encouraging signals of efficacy in the peripheral and cardiac muscle of Becker and Duchenne patients. We're currently in a phase 1 dose escalation study in biomarker and cardiac function assessments in Becker, and we're driving now towards studies in Duchenne patients. I'd like to thank all of you for your attention and thank PPMD for this opportunity to speak.
Wonderful. Thank you, Ranzi. That was a really great presentation. So this community has supported some of this early research years back, and I just wanted you to explain a bit that the term they've been used to for the years is epicatechin. So it's something that wasn't said in your presentation, but the community actually funded this early work through PPMD. So I wanted to acknowledge that, but also just have you explain a bit. Was it reformulated over time, or from the original work, or tell us how that path, or why it took to now to get to this stage? I think it'd be important for them to hear.
So, and we're grateful for the partnership with PPMD over the years. So our company used to be called Sarepta Therapeutics, and Sarepta has been around for about 10 years, and our co-founders worked very hard, and it was through the support of organizations like PPMD that we've now gotten to where we are. We rebranded the company, and we had a very successful Series A financing round in December of 2019. And yes, the core compound is epicatechin. We believe that these two stereoisomers of epicatechin minus and plus epicatechin have unique physiologic properties, as I've outlined so far. And both of these, the minus and the plus, have been studied in pilot studies before. However, we've come to realize that the plus isomer is much more biologically active and has physiologic properties that the minus isomer doesn't seem to be able to replicate, at least with the types of doses that you'd want to do in patients. So the company's been working hard on a way to refine and manufacture the plus isomer, which is extremely difficult to find find in nature. And so EPM01 is the plus isomer, which appears to be significantly more potent and significantly more biologically active. And so that's the reason why we're taking that compound into clinical development.
Thank you, Ronzi. That was extremely helpful. And, we like more potent and more biologically active. I think those are two really good things. All right, we're going to now kick this to Eric, who's going to do the Q&A of the panel. Eric?
Yeah, so just a reminder, if you have questions, please submit them to the box below and indicate which sponsor you're directing this towards. And this is one that I'm going to kind of pass around to the whole panel here. One, again, we love to see that you're all testing your products and investigating them in some of these populations that don't always have the opportunity to participate in clinical trials. But there have been some questions as well about whether these products could benefit use at a younger age, and if there's any plans to investigate these in a younger population of Duchenne. And in the case of Aperium, maybe if you have a timeline for when you may get to Duchenne. So Dr. Merrick, I will start over with you.
So yes, initially we're looking at ages 8 through 16 and looking at sort of the late ambulatory and non-ambulatory phase. And that's largely a strategy just so that we can try to, as rapidly as possible, conduct an understanding of safety and efficacy, given the fact that so many boys are enrolled in other types of studies early on. So it's for that reason. But down the road, we'll be wanting to look at the entire range, yes.
Great. Thank you. Dr. Hisham? Oh, I think you might be muted.
Sorry.
No problem.
Sorry. Sorry, Eric. Yeah, no, I agree with Dr. Merrick. Although a majority of our data is in the older population, there is no scientific reason why we wouldn't have the same or similar benefit in a younger population. Of course, it needs to be proven. We have some initial data from our phase 2 study, but certainly it's something we would look at in the near future.
Great, thank you. And I know, Dr. Markham, you indicated there's a challenge in kind of detecting efficacy with the younger ages, but is this something you still think may be able to be beneficial treating at an earlier age?
Yeah, definitely. I mean, I think if we can see a signal of benefit in the 7, 8, 9, 10-year-old who can undergo an MRI, in my mind, there's nothing to preclude going even younger. And at that point, I think you're just talking about treatment. So I don't even know if it is necessary to study it in a 6-year-old who can't do an MRI if the signal is valid.
Great, thank you. And Ronzi, if you could maybe give an estimate of when you think you might be able to look at the Duchenne population.
Well, as I said, we're working towards this now. In fact, we're looking at 2 or 3 different options. I can't make any promises, but I would love to be able to say that we would be in the Duchenne population within a year. That's certainly by this time next year. I think that's a goal that I would set for ourselves.
That's great. And again, I know there's a lot of appreciation too from our Becker population who really don't always get the same opportunities, and so I know they're all grateful for that. Mm-hmm. Dr. Merrick, so we've been talking a lot about COVID, and there was a question. Could you maybe explain a little bit more about what some of those, you talked about the at-home video and some of the remote monitoring that's being developed for the trial. Could you maybe explain what is involved in that?
Sure, I'd be more than happy to. For some of the remote monitoring, what we've done is like the things that we'd be doing otherwise, safety visits to the clinic, we've moved them to being conducted remotely by qualified nursing staff. And in terms of the video monitoring, that's sort of work that we're actually doing, looking at certain test that, of how patients are doing, where we're collaborating with Casimir on those efforts. So it will allow us maybe to bring some, I think certainly Casimir is looking at how they can bring new measures to patients. So I think everyone in the field is excited about those possibilities.
Yeah, we had a lunch and learn session that's going to be recorded and released next week for anyone interested in that work. So thank you for mentioning that. Go ahead.
Yeah, and if we just had a question come in for Aperium, although I do think we could ask this question to everyone on the panel as well. But for your study with Becker, is there any possibility about kind of expansion to female manifesting carriers?
At this point, no. We are enrolling patients with Becker and Becker-like mutations, but at this point, we don't have any plans to expand to female manifesting carriers. But I'd be happy to talk to somebody about it if there's a significant enough desire to do so.
Great, thank you. And I don't know if anyone else has any could comment on that, if their product may be possible to use in that population, or any plans. Dr. Hasham?
It's certainly possible. Of course, our trials are coming to a conclusion, so we'd have to look at the post-approval setting for any potential to do that.
Okay, great, thank you. Dr. Markham, in your in the open label extension, can you share what dose you expect patients will be receiving during that phase?
So we initially had planned an interim analysis after the 6-month interval to help guide what that extension would be. And so if there is a dose that seems to be more efficacious, then that would be the dose that would be chosen.
Okay, great, thank you. We also have had a number of questions come in just about combination of therapies. And I think that's something that we talk about a lot. It's something that we think is important. And did notice that is part of kind of the current exclusion criteria in the Astellas trial that any of the exon skipping therapies, is part of that exclusion criteria. Could you maybe explain some of the rationale behind that decision, Dr. Merrick?
Well, essentially, we are very early on. So for this initial phase 1b study, we're excluding that just because there may be some change during the study with the exon skipping therapies, and it's going to be very, very hard to be able to look at that extra variation in a very small study like this. Down the road, as we get into larger phase 2 and phase 3 studies, we'll be certainly reconsidering this strategy.
Okay, great. Thank you. And then actually, we had a follow-up question as well come in on that. Is there a plan for an open label extension in your trial?
Yes, after the first 12-week period, then all patients are allowed to go into a 12-week open label extension, yes. So also that will be available to all subjects. So the subjects that are originally on active drug will be able to get an entire 24-week period. But even the ones on placebo will have a 12-week potential exposure to the ASP 0367.
Okay, great, thank you. And this one came in for Aperium. So in your next study phase, are you planning on having just U.S. sites or looking towards a global pathway?
In the Becker or?
In the Becker trial.
Yeah, no, we would very much be looking to do a global study in our follow-on to this current trial.
Okay, great. Thank you. And so we actually are just about at time, so I'm going to say thank you to all of our panelists. We really appreciate you taking the time to be with us today. Anyone whose questions we weren't able to get to, again, we will pass those along to the sponsors. And so thank you all for joining us this afternoon. And
I'm back with I think we have a 12-minute break, I believe. Yeah, we're going to be coming up next with endocrinology and Duchenne. So thank you so much to our panelists once again, and we will see everybody in about 12 minutes.
Great.
All right.
The Duchenne Registry is an international registry for people who have Duchenne and Becker muscular dystrophy. Women who are carriers are also invited to join. A registry is a place to collect information or data about people who have a specific disease or condition. By sharing your data through the Duchenne Registry, you're joining a global effort to better understand Duchenne and Becker. Doctors and researchers use this data to improve care and develop better treatments. None of these people can do it alone. We need you. Join the Duchenne Registry through our new user-friendly smartphone app. Learn more about the app at duchenneregistry.org, where you can watch how-to videos to help you get started. Once in the app, you'll answer some surveys. Based on your answers, we can send emails about research studies you may qualify for, since many researchers use the registry for recruitment. Your privacy is important to us. Everything is stored securely. We never share your name or contact information. If you have registry questions, you can talk to one of our genetic counselors on the phone or by email. In addition to helping you with the registry, they can interpret genetic test reports and review the impact on your entire family. If anyone in your family needs genetic testing, our genetic counselors can coordinate free testing with your local doctor through our Decode Duchenne program. They can also discuss research opportunities and potential therapies. You have the power to make a difference. Join the Duchenne Registry and join the fight to end Duchenne.
The PPMD Community Resource Center is a one-stop resource for families living with Duchenne. It's a place to find some of the favorite resources, products, and organizations of our community members. Categories include education resources, independent living, insurance and funding, travel and recreation, and much more. You can search for national resources or state by state. It's a fast-growing source of information for families and was created with the help of members of the Duchenne community. To find our Community Resource Center, visit www.parentprojectmd.org/resources.
Welcome back, everybody. Next up, we're going to continue our exploration of care areas, and this one on endocrinology. So in this panel of experts, we're going to discuss things like bone health, growth, which includes height and weight, and puberty and Duchenne, as well as discuss the latest care recommendations. Helping us lead our discussion today is Kathy Kinnett, PPMD's advisor for care. We also have members of our PPMD's Adult Advisory Committee on each of these panels. So with the first discussion in this care section, we're going to discuss bone health. Dr. Leanne Ward from the University of Ottawa is going to be joined by Adam Weschler. Duchenne certainly leads to complications with bone health. Steroids can further those complications, and this is going to be a discussion on the areas of care within here, including interventions and monitoring care. We're going to have a great discussion and then lead to new discussions under endocrinology. So with that, I'm going to pass it off to Kathy to lead the discussion. Thank you.
Thanks, Ryan. As you said, there are several issues around several endocrinology issues in Duchenne that are exacerbated by steroids, and one of those is certainly bone health. So we're going to start this section off with Leanne and Adam, and we thought it would be fun to have Adam and Leanne present this information rather than me talk. So I'm going to not talk and turn it over to them.
Well, thank you very much, Kathy. On behalf of Adam and myself, we'd like to welcome you to this 15-minute excerpt on bone health in Duchenne. I'd like to start by introducing Adam to you. Adam is a 24-year-old with Duchenne muscular dystrophy living with his family in Vermont. He's currently serving in AmeriCorps as a work readiness trainer for people with disabilities. Adam graduated from the University of Vermont in 2018 with a degree in environmental studies. And here he developed a passion for sustainability and accessibility in the built environment. When he's in his free time, he enjoys reading, playing video games, and spending time with friends. Adam.
Thanks, Leanne. So now I'm going to introduce Dr. Leanne Ward. She is a professor of pediatrics at the University of Ottawa, where she has held a research chair in pediatric bone health since 2010. Recently, Leanne was the chair of the Endocrine and Bone Health Subcommittee for the Centers for Disease Control 2018 DMD Clinical Care Considerations. She's also partnered with PPMD to educate the DMD community about bone health and endocrine issues for the last few years.
Well, thank you, Adam, for that kind introduction. First slide, please. So we'd like to start by Adam sharing with you his journey with osteoporosis or bone fragility in the Duchenne context. Adam, could you tell us what it's been like for you to have fractures?
Sure. So I was first diagnosed when I was 3 years old. My parents noticed that I had difficulty keeping up with other kids my age, and I'd often fall. And so my first fracture actually also happened when I was 3, pretty soon after my diagnosis. So I think what happened is I ended up falling off a short climbing structure that was probably only about 2 or 3 feet off the ground, fracturing my upper arm. And then about a year and a half later, I had my second fracture, again, probably falling off a short climbing structure. And this time I broke my right heel. What's significant about these fractures was that both of them were before I started taking steroids. And so I started steroids when I was 6 years old. And so the following few years, from about the time I was 18 until 14, I had a number of other fractures, such as big toe, wrist, ankle, and a finger. But it wasn't until 16 that I had my first major fracture, where I ended up breaking my left femur. So at that time, I was still pretty new to using my wheelchair. I wasn't very much in the habit of wearing a seatbelt yet. And I was at a Boy Scout picnic in a park, and I tried driving my wheelchair over a swale, which is like a ditch in the ground. It turned out being steeper than I thought it would be. And the chair stopped, and I fell out. And ended up breaking my femur, was taken to the hospital, got a full leg cast, and I stopped walking pretty much after that fracture. And then at 17, as I was using my wheelchair full time, my scoliosis got worse, and by the time I was at this age, I had a 40-degree curve in my spine. So we decided it was time for spinal fusion surgery. And in preparation of this surgery, to make my bones a little stronger, I took 2 doses of a drug called pimidronate, which is a type of bisphosphonate, to strengthen my bones some. And both of these doses are about 4 to 6 weeks apart. They're infusions I had to go to the hospital for, and I stopped taking them about 2 months before the surgery. And I didn't, for the next 3 years, I didn't have any more fractures until I was about 20 years old. What happened here is I ended up fracturing both of my femurs because I was trying to drive down a portable ramp in my wheelchair as I was leaving a friend's house at college. And the ramp happened to be too short for the number of stairs, so it made the ramp too steep. And as I was going down the ramp, the wheelchair essentially tipped. I went with it, and wheelchair kind of landed on me. And so my dad was picking me up from school for the end of the semester, and before sitting me up in my wheelchair, made sure I could feel all my fingers and toes. And so once I got set up, I drove my wheelchair into our wheelchair accessible van and then drove me to the hospital. I don't really remember anything after getting in the car because I ended up developing what's called fat embolism syndrome, which Leanne will explain in a few minutes. And because of the fat embolism, I went into respiratory distress, quickly deteriorated, and had to be intubated and sent to the ICU. And so this was a very scary time for me and my family. And then finally at 22, this is where I am now. I started going to a new clinic where they identified the start of compression fractures in my spine. And so I started a new type of bisphosphonate called zoledronic acid, which only now requires an infusion once a year.
Well, thank you, Adam, very much for that narrative. That's quite an incredible journey that you've had with your fractures. I'd like to ask you a few questions. First of all, what was the most challenging part of this story for you in terms of having fractures?
Yeah, I think looking back, one of the most difficult parts was having to be aware, I guess, of my surroundings a lot of the time. And I feel like I had to be a lot more careful as a kid when I was driving around in my wheelchair. And that's often difficult as a kid because you don't always want to be careful all the time. You want to be having fun.
Mm-hmm. So you were hesitant to do things because of this fear of fractures.
Mm-hmm.
And can you tell me what it was like to receive the intravenous medications? What was that experience like for you?
Yeah, so I tolerated the bisphosphonates quite well. I had maybe only some minor side effects the first couple times I took it, such as feeling a little feverish after the pimidronate, and as well with the zoledronic acid. The first time I took it, felt a little feverish and had some digestive issues for about a day.
Mm-hmm. And I think an important message here is that it's important that these bone protection medications are given by someone with expertise in their administration so they can help you through those side effects.
Absolutely.
And my last question for you is, what would be the main message today that you want to share with the viewership from your story?
Yeah. So a lot of the fractures I've had kind of come down to fall prevention, especially in a wheelchair, because I've found that it's really important to be careful and being aware of your surroundings, especially when you're trying to drive over rough terrain, because it's not necessarily, your wheelchair may not be as stable as you think it is. And I've found that it's important to wear my seatbelt.
Mm-hmm. And I think that when we think about osteoporosis, we often talk a lot about monitoring and treatment, which I'm going to do now, but we don't maybe talk enough about fall prevention. So I think your take-home message, Adam, is really important. So thank you for that.
Mm-hmm.
Next slide. So what I'd like to do now is just to capitalize on facets of Adam's story that raise important principles about bone health, monitoring, and osteoporosis treatment for you. And so the first message is that both the muscle weakness, the myopathy, and steroids contribute to fracture risk. Delayed puberty and growth and nutrition also do as well, but the two main drivers of these fractures that Adam's described are muscle weakness and also steroids. Now, Adam talked about having a fracture of his upper arm at 3 years of age, well before he started steroids. 3-year-olds should be able to play and fall off small structures and not fracture. The upper arm is an unusual place to a fracture, and so this was a very important fracture event in Adam's story. We know that boys who are not yet on steroids can have fractures of their spine, and the risk of those spine fractures goes up that much more if they're on steroids. And steroids can also increase the risk of having long bone fractures like femur fractures, such as Adam. Next slide. So Adam talked about what it's like to have osteoporosis. I'd like to show you what osteoporosis looks like on x-rays when we go through things with families. So on the left, we see pictures of the femurs, and this is in a boy with Duchenne and a boy without Duchenne. This is the biggest bone in the body. And then on the right, we have a picture of a boy with Duchenne at different time points showing the bones in his back called the vertebral bodies. So on the left, in the boy with Duchenne, the first thing we notice is that the femur looks pale compared to the boy without Duchenne. And we know that that means the bone density is low. Certainly low bone density is a predisposing factor for fractures, but you'll also notice that the femur is smaller in size compared to the boy without Duchenne. And that comes about as a result of the subnormal mobility and the lack of weight bearing. And then the arrow is pointing to a little crack in the cortex at the end of the femur, and that's a classic type of fracture that we see in this setting, although fractures can occur anywhere along the length of the femur. And then on the right, we have a boy who's on the left of that right-hand panel, 8 years of age, on steroids for Duchenne, and he has a beautiful spine x-ray, and we've started monitoring him. He comes back a year later. He has no back pain, but I notice that he has a vertebral fracture, which you can see there with the red arrow. The bones in the back are normally very square, and they get bigger as they go down the spine. And you can see that the bone with the arrow is smaller than the one above. So I know that's a vertebral fracture. And you may say, well, that looks very trivial. And indeed, visually, in the early phases, it does look trivial. But clinically, this is very significant. It means the bones in the back have not been able to withstand the pressure from the upper part of the spine. And this is osteoporosis. The other reason this is so significant is that if left untreated, then this will just progress, and that's called the vertebral fracture cascade. Next slide. Now, Adam talked about a rare but very important complication of osteoporosis called fat embolism syndrome. And this can happen not only after a fracture, but just after a bone injury, a bone bruise, so even just after a fall, and that's very important to understand. So in fat embolism syndrome, we know that anyone with subnormal mobility and anyone on steroids can have an accumulation of fat cells called adipocytes in the bone marrow within the long bones. And then if there is a bone injury or a fracture, those fat cells can come together in the bloodstream. They're released by the injury and form fat droplets that go to the lungs, shower the lungs, and clog them up, making it difficult to breathe. And they can even go to the brain and cause problem with cognition. So this is a rare but important complication of osteoporosis, and very important to know that if you have difficulty breathing after a fall or a fracture, you need urgent medical attention. Next slide. And then finally, how do we monitor and treat osteoporosis in Duchenne? And Adam has already alluded to this with his journey. I think the key take-home message here is that we start early. We don't start after years. We start around the time of diagnosis or at latest at the time of steroid initiation. We start with bone density tests yearly, and then if a patient is on steroids, we do a spine x-ray every 1 to 2 years. If they're not on steroids, we do the spine x-ray every 2 to 3 years, and we do the spine x-rays even more frequently than that if there is back pain or if there's been a drop in BMD. And then when there's the very earliest signs of osteoporosis, including those subtle changes in the spine that I mentioned, even if there's not back pain, or if there's a single long bone fracture with little trauma, we would recommend therapy. And the bone protection therapy is exactly what Adam talked about, intravenous zoledronic acid or intravenous pamidronate given every few months, depending on the stage of treatment of the child or the man. And with this therapy, we are looking to keep back pain at bay and to keep the bones as strong as possible. And you may say, well, why not use oral agents? Because there are oral versions of these medications. And the reason is that the bioavailability of the oral agents is very low. So not much gets into the bloodstream and not much gets into the bone. And we know that IV therapy is more effective in terms of protecting the spine and helping with the bone density. So with that, next slide. On behalf of Adam and myself, we'd like to thank you very much for your attention, and we look forward to your questions at the end of this session. Thank you.
Thank you so much, Leanne and Adam. Adam, that is my favorite slide. It's the last one. I'm going to kick it right back to Ryan and Eric.
Thank you so much, Kathy, Leanne, and Adam. And Adam, I love the background of your Zoom screen. So as Kathy said, we're going to have questions at the end. Remember, there's a box right below your player. You can start typing in your questions now for the Q&A panel at the end of this. And remember, on all these topics under endocrinology, we have a number of resources on the PPMD site, some easy downloadable materials to take with you for you and your physician to have a discussion on some of these topics. And it contains information about the latest with the care considerations in terms of endocrinology. So next up, we're going to be talking about puberty. We are lucky to have Dr. Rob Benjamin from Duke University, as well as PAC member Austin LeClaire. Puberty should be evaluated each year after the age of 9 years old. And steroids may impact puberty in different ways. And this panel is going to talk about this. And this is a sensitive topic, and we really do appreciate Austin from our PAC for taking on this topic with the panel. And for all the speakers. So with that, I think I'm ready to hand it back over to Kathy to begin this next panel.
Thanks, Ryan. As you said, this is a sensitive topic, and I really appreciate Rob and Austin discussing this. So I'm going to kick it right to Rob and Austin.
Hi, my name is Austin LeClaire, and I'm 21 years old, and I'm a patient with Duchenne. I'm currently going to college for mechanical engineering, but I'm thinking about using the degree to get into bioscience. And I serve as a PAC member, as Ryan mentioned. I also am on the Duchenne certification committee for hospital locations that we want to certify to be able to treat Duchenne specifically. And I'm also a co-founder of One Rare. So I'm kind of rare in the sense that I personally haven't required any medication to go through puberty. I've been able to do it naturally, and I just have always wondered why that was. But I think it's important to know that you don't necessarily need medications to be able to go through puberty. Sometimes you need to do it, but for the most part, I think there's a chance that you could just do it naturally. And I understand that puberty is very uncomfortable to talk about, but I think it's a really important thing because sometimes when it's delayed, it can really set back your life and make it a little more difficult. But I think that it's a really good thing to talk about. And I personally was delayed. I think it came in around the age of 18. And I think the hardest part was, yes, mentally I was an adult and I was 18, but physically it's a lot different because it's hard to tell what you're going through when you're mentally an adult, because most adults don't really normally have to go through that. So I think I'm going to pass it on to Rob now.
Yeah, thanks so much, Austin and PPMD and Kathy. Austin, you amaze me with your breadth of knowledge on it already, your observations, and this continues to be the case with every young man with Duchenne that I talk with. So I appreciate the chance to talk a little bit about puberty. And I do have some slides to go through. I thought Austin and I have exchanged some emails about some questions that he thinks people may like to know the answer to. My brief introduction is I'm a pediatric endocrinologist at Duke. And I've been involved with our neuromuscular clinic here, Dr. Smith, and involved with PPMD for several years now, and greatly appreciate the chance to meet with you all and talk with you all. Austin, you have a question you'd like to ask?
Actually, I have a few questions.
Oh, great.
So I think one of the important ones is, when is puberty supposed to happen for the average person versus someone with Duchenne?
Sure, good question. Typically we think that puberty starts anywhere from 9 to 14 in boys. It's a little bit early if it starts at 9, a little bit late if it starts at 14. But usually we think of that broad time range from 9 to 14. And I'll show this in the slide too, but it kind of speaks to the variability, right? Why do some start puberty at 9 and others later than that? Another point that I'll come to is sort of getting to your question with Duchenne, and that's that medications can definitely, particularly steroids, can impact timing and how far puberty goes.
So the other question was, how do we know when puberty is happening?
Yeah,
because it's obviously varies from each person to person, but what are generally the key signs that you know it's happening?
Yeah, good question. And I'll show this in a slide as well. I think, people often attribute puberty to, body odor and hair, mood changes, though those can happen even before puberty starts. And it's hard to know when puberty is happening in your own body. There are some pretty telltale signs that we look for, namely testicular enlargement in boys. So it is a sensitive topic and one that is awkward, honestly, for patients and families to discuss. Often parents don't know when it's happened, but important to have that exam.
So I think it's another important thing to talk about is, when do you decide that you really need to start thinking about different options? Like, if you're not going through puberty fast enough, when should you decide to take hormones and get the shots for it?
Yeah, another great question. I mean, really, there's no final determination on age to do this. I think in most young men, we start at 14. We consider if it hasn't started by 14 that we'll jump in, but certainly everybody's different, and I think there are some who might think about that a little bit earlier. But I'll go through a few slides that show this. Maybe it makes sense in the interest of time to do that. Now, can we go to the next slide? So, what is puberty? Puberty is a transition from sexual immaturity to maturity. It's when a boy becomes a man or a girl becomes a woman. The physical changes are significant as puberty begins. You may notice changes in growth in your voice. This can have impact on muscle strength and bone development, in addition to every part of your body, including the brain and heart. Next slide. Why does puberty, or how does it happen? It happens because your brain sends a message to your testicles, and your testicles make testosterone and sperm. The exact messages aren't important. I put the letters up, but I come back to the slide as we talk about how steroids and testosterone can impact that. Next slide. I bolded testicular enlargement because, as I said before, this is really the sign that it's started. All the other things that number one in particular, hair in the private area under the arm with acne and body odor, can be associated with the beginnings of puberty, but also can happen before that happens. And then after you get into puberty is when you make the male hormone testosterone and sperm. Can we go to the next slide? as we said, when it usually starts from 9 to 14. It used to drive me crazy when girls always told me they were more mature than me when I was 9 or 10, but it's true that girls start before boys. They start about 2 years ahead. So, because a younger sister has started and you haven't, doesn't mean that you're late. And that's an important point. It's on average 2 years later for boys to start. And boys usually have a longer pubertal time than girls. Next slide, please. We typically say that puberty is late when when nothing has changed significantly by 14 years of age. And you mentioned this, Austin, but others may wonder, why haven't we noticed this even the late teenage years? And partly it can just be your body. It sounds like your body wasn't ready to do it. There's a wide variety of ages when we hit our growth spurts, for instance, or when other changes happen. So not everybody is supposed to do it at the same time. But there's another big reason why this might happen in Duchenne. Can you go to the next slide, please? And that's steroids. And I apologize at the outset for misspelling Emflaza. It should be an M, but steroids can block the brain message. That's really their main problem with what they do with puberty. So those two abbreviations I showed you before get blocked by this high dose of steroids usually, and that can lead to then a block in your body going into puberty, testicular enlargement, and sperm production. Next slide. You asked when we start testosterone or when to do things, and that's, again, usually 14, though we may attempt to jumpstart puberty before that. What do I mean by jumpstart? We may give a short course of testosterone because we'd always rather that you do it and not get it through medication. Yes, Austin?
Yeah, so I think it's really important to also consider, I know one of the main concerns that many parents have is putting their kids through puberty too early or don't want to do it at all, because when you're going through that, you can be a pretty big jerk and be really nasty to people. Right. So I think maybe it's also a consideration of how behaviorally, how will it affect their behavior? But obviously that's a natural thing. But I think when you have to do it medically, I think it generally affects it even worse than normally.
No question. I think, as you say, that medicines and interventions can have those effects that we don't want. And that is part of changes that happen with puberty or mood changes, but they can also happen before puberty begins. So moodiness doesn't mean puberty has begun. Anytime you put somebody on medication, like testosterone, we have to think about those side effects. And again, we want to make sure you can do it before we step in to help with it. Jump starting it and giving a short course is fine and no problem. But more long-term treatment with testosterone is usually we try to wait and see if your body can do it first. I don't know why some have it as late as they do, but that's why seeing your doctor regularly and having those exams and those conversations are really important early on. Like I said, it's an awkward topic for patients and for families and for physicians, but it's a super important one to have. I think you've asked, my other slides show essentially what you've asked, so I don't know that we have to go through it in more detail, but it's just I think some of the take-home points are that it's important that your body make or you receive testosterone because we know it has important effects on bone health in particular. Can also help with muscle strength, but really in patients, particularly young men with Duchenne who are on high doses of steroids, this can be an important bone protector. As Leanne talked about, we don't have a lot of other options for bone treatment, and so this is one that if we think you're missing can be helpful. Do you have any other questions, Austin? Did I answer?
I think the final question was, why do some people have normal puberty with Duchenne and some have late puberty and then some don't have puberty at all?
Yeah, good question. Well, I think those who typically go through puberty often have a different need for steroids. I think steroids are the common theme. We talk about bone health, and you'll also talk a little bit about growth. Some of those can be impacted by Duchenne without steroids. Puberty probably isn't. If you don't get put on steroids, you probably will go through puberty on your own. And so it's really a medication effect as much as anything. It's why I don't always like the term delayed puberty in Duchenne. I feel like it's blunted in many young men. So probably medication, the dose of Emflaza that you're on, and then, as we said, just timing of your own development. Probably all of those play into it.
Thanks, both of you guys. This is such an interesting topic. And Rob, I just have one question. It seems when you talk to families that the endocrinologists sometimes are reluctant to start this conversation around puberty and around testosterone. Are there reasons for that, or do you have an age that this conversation should start?
I'm biased, but I think it should start with our first visit, just to talk about what we see. I think it's probably the awkwardness of that topic. It's a sensitive topic and an emotional topic. The idea that you may not go through this on your own. That's that can be traumatic for families. So I would imagine that might be one of the things preventing that conversation from happening. But I think we try to have that conversation on day one, even with a young patient. Talk about all three things: bone growth and testosterone and puberty, because that way it helps people think about the future and what we can do.
I think that some of the issues come when they are seen at a center that maybe doesn't have an endocrinologist that routinely sees patients. If that happens, is there an age at which parents should request that they see an endocrinologist or have that conversation with their neurologist or neuromuscular specialist?
Yeah, I'm biased on that answer too, because I feel like I think endocrinology has a role as soon as diagnosis happens, and so I would be in strong favor of having some connection with a pediatric endocrinologist who's involved in clinical care right after diagnosis, or as soon as possible after. If it's a burden for the family to travel, one thing we've learned with our current health environment is we can do things remotely. We can connect with families, and through groups like PPMD, we can make those connections even if we're not there in person. So I would advocate for that, anytime.
Do you know if there have been studies that have demonstrated the advantages of testosterone or the impact of testosterone on muscle strength and bone health? Or are those just assumptions? I know they've been a little bit. And in Duchenne?
I know Leanne has more breadth of background and with that, and may be able to speak to it. Maybe we could come to that in the Q&A at the end. I think there's still a lot that's unknown, but I think it is pretty clear, pretty established that the testosterone, or the absence of testosterone, is a risk factor for bone problems. So I would support that, or I think that's supported in literature.
Okay, thanks. Thanks, both of you. This is really fascinating. I'm going to send it back to Ryan and Eric in the studio.
All right, thanks so much, Austin. I appreciate it.
Yep.
Thank you. That was a really, really great discussion. And I think it gives a lot for parents to be able to go, and families to be able to go to their care teams and have a discussion about everything that we talked about over that last panel. And again, the care team at PPMD is at the ready to talk to you prior to those appointments. I think the take-home message of having these discussions early on are so critical. And, let's face it, some of these issues really do impact the, social, emotional health of the people we love with Duchenne. So we should not shy away from them. So our next panel is going to be on height and weight. So we're joined by Dr. Phil Zietler from Children's Hospital of Colorado. And we also have Colin Worth from PPMD's PAC joining the conversation. And I'm going to hand it over to you to start this discussion on height and weight. Kathy?
Thanks so much. And welcome, Phil and Colin. We're so glad that you guys are here to talk about this. This is one of the most discussed topics on social media, so I will turn it over to both of you to enlighten us.
Okay, thank you, Kathy. Nice to see you, Colin. Colin and I have talked a few times in planning for this. And thanks to PPMD for allowing us to chat with you guys. We're going to start with just a couple of slides to just orient folks about sort of typical growth and what is generally expected. And the key for understanding this really is to understand growth charts. And this slide here gives an example of different kinds of growth charts. On the furthest left is a chart that looks at growth between 0 and 36 months of life. That's designed for for babies who are laying down. And then this middle chart here, which happens to be for girls, but the same ones exist for boys, is for 2 to adulthood. And this is really designed for tracking the growth of people who are standing, individuals who are standing. So it's important to keep in mind that this is really intended for standing height. The way this was done was collection of heights and weights among middle-class white kids in the United States, essentially, and then putting together what this looks like cross-sectionally. So this was not the growth of people being followed over time, and it doesn't include the growth of people from various racial and ethnic groups. So it's important to keep in mind the limitations of those kind of growth charts. There are longitudinal charts where the same people were measured over time, and that allowed the generation of growth velocity charts, like you see here on the right. And what you see here is that growth velocity falls during childhood, being very rapid at about age 2, falls during childhood, and actually reach its lowest point right before growth begins to increase at puberty. So you can see that if that pubertal growth spurt is not normal or it's delayed, like Rob and Austin were just talking, that's going to have an impact on the way in which growth is going to look on other charts. Can we see the next slide? The other thing we need to talk about quickly is bone age evaluation. This is a common part of evaluation, and this gives us the ability to look at the skeleton and compare it to standards which have been put together of what the skeleton looks like and what the growth plates look like in an average person of a certain age. And that allows us to track how quickly or slowly an individual is growing. And we take that into account when we're looking at growth also. Next slide. This gives an example of that. So here's the growth chart of a person who's kind of short, but in fact, they're growing more slowly. They're developing more slowly than typical. So their bone age is delayed, not a 14-year-old, but that is of an 11-year-old. That means that they have the growth potential left of somebody who is 11, not 14, and therefore they're going to be taller than it seems. So the endocrinologist can actually make a prediction about what the final height's going to be. And in many cases, such as in delayed puberty, like you see in boys with Duchenne's, that they may in fact end up taller than it seems because their puberty is delayed. And in this case, this girl's height prediction is appropriate for her family. So those are three things important: the growth chart, bone age evaluation, and taking into account what the growth potential actually is. I think the next slide has to do with Colin's own story, I believe. Yes. All right, Colin, would you like to tell us about yourself?
Hi there. Thank you for, I'm glad to be a part of this panel. We're going to talk about two things today, both my height and my weight. So the one thing, I I will note is I started steroids, prednisone at age when I was almost 10, and I was on that for around 5 years, and I had issues. As you see, my height had started to go under the normal curve, and also I had gained a lot of weight, and then I had started seeing the team out at Cincinnati Children's, and I ended up switching from prednisone to deflazacort, and I also started metformin to help me to lose weight. Well, first I'll talk about height, and then Phil and I can talk more about the weight issues. So as you see, I was around, I started growth hormone in the end of 2010 when I was almost 16, and I was on it for a year. I stopped in the beginning of 2012. So my height it definitely started to, my growth definitely started to become slower after I started steroids. And you see, when I started growth hormone, I was around 145 centimeters, or 57 inches, which was below the normal stature for someone of my age. And but Phil and I had talked about this, but as you see, after I was on the growth hormone, or while I was on the growth hormone, it didn't make much of a difference in my height. And Phil and I had discussed this, that actually, after looking back, having done it myself, I don't know if I would have taken the growth hormone, because it didn't really make much of a difference. And it was also, I stopped after a year because there was, you have to take daily, I had to do daily shots with growth hormone. So I kind of got tired of that. But it didn't really make much of a difference. And I think also, being in a wheelchair, height is, not the most important thing. People don't notice your height. And also, it's easy, I think it's also kind of practical because someone in a wheelchair, and also being smaller, it's easier for caregivers or parents to handle you, especially if you're non-ambulatory. It's definitely easier if you're smaller for transferring. I don't know if you want to say something.
So, yeah, I just want to say that I think it's important, and this is something you and I talked about, and that is that lots of times people get very hung up on height, but it's important to keep in mind what the optimal height is for somebody, and that may be something quite different for an individual who's in a wheelchair and requiring a lot of physical care than it is for somebody who is otherwise ambulatory. And I think for a 15-year-old guy, it's easy to feel like height is really important. But I think your recognition that maybe we need to be very careful about that message and whether or not height really is something that we should be pushing very hard. Do you want to? How about let's talk about your weight and we only have a couple of minutes, but I think it's really important. So tell me, you started metformin for insulin resistance and to cause weight loss. What do you think was, you lost a ton of weight, Colin. What was the trick?
I think I definitely think the metformin helps, but I mean, I've always been very watchful over my diet, making sure I don't eat too much. I'm avoiding carbs and sugars. That's important to make sure you're at a healthy weight. And I know it's hard being someone in a wheelchair, you're not burning calories, but trying to limit your caloric intake. I mean, I know, it's hard to say don't, guys enjoy eating, but everything in moderation is okay. And I really, I've tried to avoid especially sugary drinks, what we call, or saying empty calories, so calories that really getting a lot of calories from something you don't necessarily need. And it's also being making sure you're not overweight is good for your overall health, including obviously if you're it's good easier on your heart too. So and also it's again easier for you to be handled as well. So since I'm of smaller stature and I'm not too heavy, I'm able to do transfers without a Hoyer lift. So just it's easier all around with good weight. I know it's hard to avoid diets, but it is possible if you're persistent over it.
So I think you've done actually an amazing job. I wish people could see your chart better. I don't think it's very obvious, but Colin managed to bring his body mass index down from really quite high back into the normal range. The issue of metformin. Metformin can be helpful, but I think it's really important for people to understand that this is not a miracle drug. The average weight loss with metformin is on the order of 8 to 10 pounds. So this is not going to make the difference between obesity and non-obesity. However, there are data that indicate that in people who do make lifestyle changes, metformin can accelerate the amount of weight loss in those people. So it doesn't do it much by itself, but if you are making changes, in general, people will see a greater degree of weight loss with metformin than without it. And then more recently, I know that there has been some discussion of the potential role of metformin to promote muscle development and potentially prolong ambulation. Metformin is a very safe drug. It's very inexpensive, etc. But the effects of it are pretty limited by my view. So this is not magic.
Thanks to both of you. The metformin discussion is one that I'm sure will come up again in the question and answer period, as I'm sure you would expect, Rob or Phil. So thank you so much to both of you. I'm going to turn this back at this time to Ryan and Eric, and then we'll get ready for the Q&A part of the discussion. Ryan?
Great. Thank you so much, Kathy. Another incredible panel. So as we're getting set up for the Q&A, hopefully you're submitting your questions into the box below the player. This is an easy way to get your questions and comments to now what will have all three of these sessions come together as one, so we can go across and ask questions in a number of the areas that were discussed under endocrinology. Looks like the panel is getting ready, and we're nearly there. One quick plug, we are going to be launching in the coming weeks a survey for the community on steroid patient experience, and a lot of that experience is linked to steroids. This is the first time the community has ever really been surveyed about how they think and feel about the use of steroids. This has been standard of care for so long, but we've never done preferences on that. So more to come on that now. I'll hand it back over to Kathy, and she will start the panel on endocrinology Q&A.
Thanks so much, Ryan. We have a number of questions, as I'm sure you would all expect. So I'm going to start with bone health. Austin, Adam, I'm sorry, did say that you'd had some side effects when you had your first dose of bisphosphonates. So Leanne, I know that experts should be giving bisphosphonates. Would you recommend that the first dose is given in the hospital? And what's the side effect profile generally like after the first dose?
Mm-hmm. So the side effects are most significant with the first dose, although in my experience treating boys with Duchenne, sometimes after the second and third doses, there can be some side effects as well. The side effect is an acute phase reaction. It's like an inflammatory response to the drug. And so what we see about 12 to 24 hours after giving the first dose intravenously is low-grade fever, aches and pains, and often at the site of, the back where there might have been pain to begin with. And that can be really quite unsettling for people to feel that degree of aches and pains in the muscles and the bones, and also tummy upset and sometimes vomiting. Now, we give medications to completely shut that down to the best of our ability, but certainly this can be quite unsettling. I think it's really important for families to know that this might be bumpy with the first infusion. Many years ago, we used to admit all the patients on these medications, and not just boys with Duchenne muscular dystrophy, to hospital, but it really was just supportive care that we were giving. And so in the end, we decided to do it as an outpatient. And I think that as long as you have really explained to the patient what to expect, and you've given them the appropriate medications to handle those side effects, and importantly, you give them an on-call number to call if they have concerns, then really it goes quite nicely. Two other important things to mention are that, first of all, with fever and vomiting, that can interfere with the steroid therapy. They may need more steroids because of adrenal suppression. So sometimes to help the boys feel better when they have these side effects, I'll give them some extra steroid to get them through that period. Sometimes there can be a drop in the calcium in the blood as well. And we don't test for that anymore because it typically doesn't cause problems, but we give extra calcium after the infusion as well.
Thanks so much, Leanne. Colin, did you have some thoughts on bone health?
Yeah, so I've had experience, I guess, with everything, with issues from being on steroids. In addition to height and weight, I've had some bone issues as well, and also testosterone. I can speak to that in a bit too, but just with bone health, I've had some vertebral fractures, and also a few years ago, I broke the head of my femur, and so one of the things I wanted to say, Leanne had mentioned that the IV bisphosphonates are better absorbed than the oral. I was actually on the oral Fosamax for a while, and I think that actually was having side effects with reflux with that too. But I've switched over to doing the Lendronate, and that seems definitely, even after my first dose, I noticed dramatic improvement in my bone density score. So yeah, I think definitely the IV bisphosphonates are certainly a better option, as you explained.
Mm-hmm. Thanks, Colin. It does seem, Leanne, like some people will start with the oral bisphosphonates and then move to the IV. Is there thoughts that you have around that, or recommendations that you have around that?
Mm-hmm. You know, my concern with that approach is simply that once you make the decision to start bone protection therapy, it's because you're seeing evidence that the boy or the man with Duchenne has lost bone strength. And so I think you want to move in with something that is, you know, more potent. We don't have that practice at our institution, and generally in pediatric osteoporosis conditions, not just steroid-treated Duchenne or steroid-naive Duchenne, but also osteogenesis imperfecta. Globally, I think most centers would initiate therapy with intravenous agents. I think one of the questions that comes up is whether after you've really stabilized the osteoporosis and helped with the back pain, like Colin mentioned, and you've treated for many years and things are really nicely stable, the individual has stopped growing, which is a risk factor itself for fractures. Is there a role for oral agents, you know, later on? And I think that's an unanswered question, and it's one that I would be open to. I think at our center, we do once yearly zoledronic acid, like Adam talked about. But that is a question that people ask me. What about oral agents once things are stable later on after someone has finished growing? And that's an unanswered question at this point in time.
Thanks so much. There are so many unanswered questions, aren't there? So many things we're trying to discover. Rob, one of the questions that comes up is how long do boys necessarily need to be on testosterone? Is this a lifelong drug, or is there a point at which it can be stopped?
A good question. I think, you know, the human body makes testosterone for most of our life. So if you commit to starting it and there's, you know, no change in therapy, you're still on those high doses of steroids, and you've decided that it doesn't look like the body can make it on its own, then yeah, I think you're on it for good. If there's a steroid change, and I know a lot of research is being done to look at better steroids that could give you some of the benefit that we obviously want to see without those side effects and costs. You know, so if something changes in therapy, or you change dosing regimens, then there's no harm in challenging the body by stopping it and seeing if you're able to do it. You just have to give it some time, because your body will not be used to, you know, having to do it if you've been receiving a medicine for it.
Thanks. In addition to puberty, is there an advantage of growth with testosterone, or would you expect to gain height like you would during normal puberty with testosterone?
It sort of depends on how you're growing before you started, I think. If you know, steroids have such a whopping effect on growth, that my experience has not been that testosterone turns that around very much, and it's again just because that strong impact that steroids have on the bone. I haven't noticed a big turnaround. I'm always optimistic for that. You know, we do start slowly and progress slowly with testosterone therapy, but we pay close attention to height, you know, after starting it, and I haven't noticed much growth improvement in most young men starting testosterone.
Thanks. Phil, there are a lot of questions about growth hormone that are coming in. So is there a time that you would start growth hormone, or an advantage to starting growth hormone earlier, or should it be done in conjunction with testosterone, or what are your thoughts around that?
So in general, the earlier growth hormone is started, the better the outcome's going to be. Now, most of that is work that's been done in other situations, and as the theme of this whole hour has been, steroids are a problem, and whether or not starting earlier will really make a huge difference in the setting of high-dose steroids. I think that's somewhat unclear, but I would say in general, the earlier you start, the more opportunity there is to have an impact of growth hormone. I think one key question or one key point, though, is if it's not making a difference, consideration should be given to stopping it. So it might be reasonable to start it early, see if it's going to have any impact at all. But then the downside of starting early would be just being on it for a really long time and having no impact, and that's expensive and burdensome. So I think if you do start early, you'd want to say, okay, we'll use it for X period of time, and if we don't see a difference, we're just going to stop.
So what would your definition of early be, or would that be unique to each individual? And also, how long would you recommend that it's given before it's reevaluated?
Yeah. Well, it's going to be somewhat individual, but I would say as soon as you start seeing growth deceleration would be the optimal time to do it, because if you can prevent the height deficit early on, that might be better in the long run rather than trying to recover from a deficit that's already established. And then as far as how long, probably 9 to 12 months on the outside. If you're not seeing anything at that point, you're not going to see much.
Okay, thanks. Leanne, are there impacts of growth hormone on the bones that we should be aware of?
So no one's actually looked at that formally in Duchenne muscular dystrophy, and even testosterone and its effect on bone is unclear. So we know that outside of the Duchenne setting, that both growth hormone and testosterone exert a positive effect on bone mineral accrual via muscle. So the big question mark in this setting where you have a myopathy is whether that same growth hormone, testosterone, muscle bone positive effect will be there because of the underlying muscle condition. No one has done studies where they've given half of boys on steroids with Duchenne testosterone or growth hormone and not the other half, and then looked at how those kinds of outcomes evolve in terms of growth and effect on bone density. So frankly, we really don't know at this point. We do know that most boys with Duchenne make adequate amounts of growth hormone, and that it's really the effect of the steroids largely directly on the growth plate. It's very hard to give enough growth hormone to override those effects of steroids on the growth plate to optimize growth in this setting.
Thanks. Phil, we've had a lot of questions about diet, and some centers recommend specific diets, low glycemic index diets, other diets. Is there anything that you would recommend that has been helpful for these patients? Anything Duchenne specific? I haven't found anything, but I wondered what you have found.
So it's, I think diets are like cold medicine. If there was a good one, there'd only be one. Right? So the reason there's so many different diets is because there isn't a magic diet. The fact is that the best diet is the one that that fits with the person's lifestyle, that fits with their preferences, that's something they're going to stick with. So it is true that a lot of people end up just eat too many carbohydrates naturally. So cutting carbohydrates can be very effective. It's very concrete. That can be useful, but there's nothing magical about the carbohydrates. It just tends to fit easier in people's lives and their dietary patterns. So whatever somebody's going to be able to stick to is going to be the most effective kind of diet. There's no science that suggests one is otherwise better than others.
Thanks so much. And I guess now that we've all gained the COVID-19, we'll have to all come to you with your diet recommendations. So with that, I'd like to thank the panel. This has been really fascinating. I appreciate everyone's input and your participation. And with that, I'll send it back to Ryan and Eric in the studio.
Thank you, Kathy, and thank you, panelists. That was an incredible discussion. We really, really appreciate it. I know a lot of questions were coming in, and needless to say to the community, we will come back and address many of them after the meeting. So we're going to take a break. We're going to come back at 5:20, and we're going to take a closer look at acute pulmonary management in Duchenne. So we will see you at 5:20 p.m. Eastern Time.
Hi, my name is Allison Joseph, and I have two sons that were diagnosed with Duchenne muscular dystrophy, Hunter, age 5, and Noah, age 4. Shortly after diagnosis, I found PPMD's Endurance Fundraising Series, and it's truly had a great impact on my life. Being able to run and raise funds for PPMD has given me an incredible sense of hope, a way to relieve my stress, and also has given my family, friends, and our community an opportunity to help us fight Duchenne. The nice thing about the Endurance Series is there truly is a race for everybody, and they're all across the country. You can sign up for major marathons such as Chicago and New York City, or do shorter distances such as the Shamrock Shuffle 8K right here in Chicago. To get more information or to sign up for a race, go to www.racetoendduchenne.org. Together, we can end Duchenne.
Hi, my name is Ann Martin, and I'm a genetic counselor at PPMD, and I direct the Duchenne Registry and the Decode Duchenne program. For those of you who don't know, Decode Duchenne provides free genetic testing and counseling to patients and families affected by Duchenne or Becker, and this includes free carrier testing. Genetic testing is so important for Duchenne for several reasons. Of course, for accurate diagnosis, but also for family planning and carrier testing of the mom and other females in the family. And equally important, knowing your mutation is crucial for understanding if you may benefit from mutation-specific therapies such as exon skipping and nonsense mutation read-through. I want to thank our industry sponsors, both Sarepta Therapeutics and PTC Therapeutics, who generously fund the program. Decode would not be possible without their continuous support. Since we launched Decode in 2013, we've provided over 6 years of free genetic testing to families in the US and Canada. Over 425 clinicians have used our program. Over 250 women have had free carrier testing in the past 2 years. And over 1,200 patients have received diagnostic confirmation of Duchenne or Becker. So if you think you or your loved ones could benefit from Decode Duchenne, please visit our website or reach out to us by phone or email. We have 2 board-certified genetic counselors here at PPMD, and we're always available to help answer your questions.
Welcome back. We're going to start this session with some polling and a pop quiz. If you haven't done this before, you need to go to pollev.com/PPMD. There's a link below the player for you to participate. Just something to remember, this polling is for our families only. The first question should appear in just a minute. You also have to type in your screen name. We need your full name that you registered with, because after these polling questions, we're going to do a pop quiz, and there's some fun prizes associated with them, but we need your full name. So you can always edit that screen name after the fact, even after you take these polling questions. So first question, current steroid regimen. This is, we placed this here because we just had a series of talks on endocrinology. Steroids came up a number of times. We've asked this question before in polling, but we're just interested in understanding what your current regimen is. We see a lot of daily deflazacort and Emflaza as the highest choice.
Yeah, and we know there's a lot of different ways that these are prescribed and taken, and so it's kind of a great way for us to capture what we see in the community right now.
Mm-hmm. So we see, for the most part, the majority being deflazacort or Emflaza, and that's followed by prednisone daily. We have still coming in some who never use steroids, some who used to use steroids.
And as we discussed in some of in the therapy pipeline, that use of steroids can have, changes on what trials you may be eligible for. A handful of trials that look at patients that are steroid naive, and then a lot of our trials that have, that inclusion criteria that does require a steady dose of steroids, whether that's weekend or daily.
Great. I think we can move to the next polling question. Here, we're talking about who checks your child or your weight. We understand that different people within your healthcare sort of ecosystem could be doing this, a healthcare aide, your doctor, your primary physician, a number of different people. So you could choose all that apply among this list. You see Duchenne specialist or physician.
And this may change too over the course of the progression of disease, and whether or not you need some assistance with a Hoyer, or with the little ones it might be a touch easier.
Someone in the household has a high percentage there, and some don't monitor weight, and that's okay. We just want to get a sense here. None of your screen names are made public. There's no judgment here. It's good to see sort of the spread of variability in the community of how weight is checked and who's checking it. So.
And just a reminder, as we talked about some of those endocrinology issues earlier, especially some of those that can be, difficult subjects for teen and adults to kind of have those discussions with their parents. We do have that Zoom meet tonight for those teen and adults. It's private, can kind of have those discussions around, what we discussed in some of those earlier sessions today.
Okay, we can move to the next question. So here we want to know how often is your or your child's weight checked? We got weekly, monthly, every 3 months, yearly, twice a year. Not sure, don't really keep track of that. We see twice a year coming up quickly in the answers. Many of our families do 2 visits a year to the neuromuscular center, and that might be when they're getting that weight checked.
And it might depend, again, this may be a reflection of whether you're doing this at home or whether this is taking place through, the home health aide or a specialist. It's definitely going to have an impact on how often you're going to be checking weight.
Sure, and then the other factor there is insurance. We have an approved steroid with Emflaza. Some insurers might require you to get your weight checked because of the policy for the insurer on continued use. So lots of different reasons here. We see that twice a year is the most popular answer. Let's move on to the last question. This is the pop quiz. So this is my favorite question. Big Simpson fan myself. What is the name of the police chief on the TV show The Simpsons? We've got several different answers coming in.
I will say this was, of the pop quizzes that Ryan showed me, I think this may be the only one that I got right off the bat.
These are all great police chiefs, I must say. There's, you know, even though there's one answer, great police chiefs listed on this list. A couple of them cartoon characters, and I'm a fan of cartoons. So we will give a pop quiz prize at the end of the day. We are going to announce who that winner is. I will reveal the answer. It was Clarence Wiggum, or Clancy Wiggum. Please stay tuned. The person who answered this the quickest will be awarded a prize, and we'll announce that a little later on. And remember to edit your screen name to make sure it's your first and last name, what you're registered with. That's the only way we can find you and provide you that prize. Okay. So with that, I'm going to kick it to Eric.
Thanks, Ryan. So next up in our care series, we are going to be talking about acute pulmonary management in Duchenne. So we all know respiratory health is a critically important aspect of this disease. And so in this discussion, we're going to talk about some of those early interventions that we can be using. We're going to talk about how to protect the lungs and maintain pulmonary function. We're going to discuss creating a plan for potential emergencies. And then finally, we're going to look at what we've learned from the COVID-19 pandemic so far. So leading this discussion from PPMD is Rachel Schrader, our Senior Vice President of Clinical Care. And if you have questions while this session is going on, please remember you can put them into that box below, and we'll address them when we get to our Q&A. And leading off for us in the first discussion is going to be Dr. Richard Schell from Nationwide Children's Hospital, who's going to be talking to us about prevention. So, Dr. Schell, you can take it away.
Sorry about that. Thanks, Eric. We miss you here at Nationwide, but I am thrilled to be able to talk to you guys today about respiratory care and Duchenne muscular dystrophy. I hope to give you an overview of what the doctors and nurses in the clinic might ask you about respiratory health, as well as discuss some of the procedures that we may recommend. This all comes from the consensus guidelines that have been published and are available as a link on the PPMD website and other websites as well. But then we'll move into more acute emergency care and finally talk about how COVID has affected this overall care. So next slide. So this is probably my most important slide if you don't take anything away from this session. Multidisciplinary care is the key to the care of patients with neuromuscular disease. It takes many, many different experts to help keep everyone healthy and safe, both in the clinic and out. And the ability that these specialists have to talk to one another also adds to a benefit of this type of clinic. And I've just listed many of the people that you may see in a multidisciplinary clinic. And if you keep listing all of them, then it gets too small to see. So next slide, please. Remember, we have a lot of muscles in our bodies besides the big ones like our arms and our legs. The group of important respiratory muscles are shown here, and they include the diaphragm and the abdominal muscles, the internal and external intercostal muscles that are between the ribs, and a group of accessory muscles that come into play when we're having more trouble breathing. These muscles get weaker in Duchenne just like the leg and arm muscles do, and we need to make sure that they get the full attention and support. Next slide. There are 4 respiratory stages that we see in Duchenne muscular dystrophy as patients grow older. Initially, everyone has essentially normal breathing and function. In the next stage that occurs in the early teens, we start to see some impairment in cough, but some of the respiratory muscles can remain strong. It's important to follow pulmonary function closely here. By the mid-teens, we have normal breathing in the daytime, and sometimes the breathing is more affected when you go to sleep. We call this hypoventilation, when you breathe more shallow, as our muscles are having more trouble taking a normal breath. This stage happens around the time that you may need to fully use a wheelchair to get around. We might recommend a sleep study, but hopefully we will get to this, but hopefully before we get to this point. The final stage happens when support is needed during the daytime as well. There are therapies and devices that we may recommend continuing to support through this stage as well. And these stages do mirror our decline that we may see in pulmonary function. Next slide, please. When a patient is about 5 or able to do the test, we will start practicing pulmonary function testing so that as they grow, they'll be able to function and we'll be able to pick up early changes in their lung function. It's a test that we try to make more fun at times by blowing out candles in the younger crowd, but we do know adults like to have fun as well, and we will let them do the same incentive spirometries for us. We'll use some nose clips and a mouthpiece to make it as comfortable as possible. I've listed some of the values that we follow, and you might hear your pulmonary doctors talk about some of these. In clinic, I tend to focus on the forced vital capacity, or the FVC, which is the maximum amount of air that you can blow out from your lungs after taking the biggest breath that you can. Another important value is the cough flow, which helps us to estimate how strong the cough is in helping clear secretions from the lungs. Most clinics should be able to do this testing when you're there for follow-up visits, and if not, you should really ask why. There are no dumb questions ever, so if you don't fully understand the conversation about lung function, don't be afraid to ask. Next slide, please. When the cough flow gets too low, we will recommend using a cough assist or teach patients and families a more manual way to help clear secretions from the lungs. This therapy should be like brushing your teeth every day because I believe it is a preventative therapy, but also a therapy that can help clear secretions when you're sick and prevent pneumonia. It basically helps you take a big breath and then it sucks it back out again to help you have a more effective cough, kind of like a vacuum cleaner in the end. I am a huge advocate of this therapy, and for those of you who are listening to me right now who have one at home collecting dust in the closet, I urge you to take it out and use it. Next slide, please. So I'm a little disappointed because I had some animation here, but chest therapy or clearance is the most important preventative therapy that we can do when our muscles are weak and unable to clear all of our secretions. And I really look at these devices and I use the analogy that they're like a wheelchair for our lung muscles. So I'd say next slide here, and the wheelchair would move off to the right so everybody can see that. But another device that we might also recommend, or you might have heard about, is something called a therapy vest that basically shakes the muscles, the mucus loose. This was developed for patients with cystic fibrosis. There is not great evidence that it works well in Duchenne because if you shake it loose but you can't cough it out, it's still down there causing problems. This therapy may be most beneficial for a patient who has had issues with recurrent pneumonia, but my belief is that it should always be paired with the cough assist. Next slide. Another concern that we may need to monitor for in the later stages is scoliosis. Significant scoliosis can certainly affect our breathing at times. There's evidence that if spinal surgery is done when needed, quality of life and longevity is improved. This is further improved when breathing support at night is added as well. It is very important to discuss this with your team and plan a surgery if needed with good communication between the team that will be taking care of things in the operating room as well as in the hospital when you are recovering. Here at Nationwide, we have a neuromuscular pathway that includes the Duchenne, our Duchenne team, along with orthopedics, anesthesia, and our intensive care docs as well. Next slide. Sleep studies are needed when symptoms listed here become more evident. Your team should always be asking about sleep-related issues at each visit, and if they do not, you should bring it up. Behavioral changes, waking up at night more, feeling tired during the day, snoring, and headaches can all be signs of disordered breathing when you're asleep. We may also suggest a sleep study when pulmonary function declines, even without a lot of symptoms, so we don't want to miss anything. Sometimes, depending on the clinic, we may check a carbon dioxide level during pulmonary function testing as another way to just double check that we're not missing some symptoms that may not be presenting in clinic. Next slide, please. If the sleep study shows that a patient with Duchenne needs more support at night, we may talk to you about the use of non-invasive ventilation or BiPAP. It's non-invasive because we support breathing with a device that helps you take a more normal breath through a mask. BiPAP is the preferred method of ventilation in Duchenne as compared to CPAP, as BiPAP helps both with inspiration and expiration. Invasive ventilation with a tracheostomy tube is a choice some patients make when they are using BiPAP for longer and longer periods throughout the day. This is a very important discussion to have with your care team, and there may be some other options as well, but unfortunately we don't have the time to really delve deep into that right now. Next slide, please. In summary, or my bottom line, multidisciplinary care is essential in Duchenne muscular dystrophy. If we don't ask about a specific breathing issue, please bring it up to us. Get pulmonary function testing at least yearly. Watch your weight and nutrition. Discuss any concerns you have about back pain or changes in your spine. Realize that sleep issues are important to talk about and to treat. Do your chest therapy that's been prescribed and have an emergency plan. And that emergency plan is going to be talked about by Dr. Meyer next in a little bit more detail. But I'm going to end my talk here, and I'm going to turn this back over to Eric.
Thank you, Dr. Schell. We really appreciate that. That was a lot of great information and some really helpful tips for what some of these families can be doing. We're going to move on to our next speaker, which is Dr. Hank Meyer from the Children's Hospital of Philadelphia, and he's going to be talking about emergency care planning. So Dr. Meyer, I'll hand it off to you.
Wonderful. Thank you. So I'm Hank Meyer from Children's Hospital of Philadelphia, and have been involved in our neuromuscular program for many years. Should the slides be up on my screen? I see them in the small screen at the bottom. Anyways, we'll go to the next slide, please. Sorry, the way it's presented, I just see me on the screen and the slide is in the gallery view at the bottom and I can't read it. Is there a way to make the...
Hi, we are just going to figure out the slides for just a bit. And I think he's got it. And we're going to go back to Hank Meyer.
Okay. Now what happened? I'm not seeing the slide. It's down at the bottom and it's not on the active screen. So here, I'll just do it manually from my computer.
All right, so we are going to take one more second just to see if we can sort this out. This is kind of the challenges that we do face when we go to a fully digital panel. So hopefully we can fix this and be good to go.
It's a great panel so far.
Yeah. Really important subject matter. So we're grateful for everyone who's able to take their time to talk to the community about this.
Absolutely. Absolutely. We heard from companies earlier today that we're focusing on pulmonary function, which is, it's so great to see drug development in that area. It allows for a broader inclusion of patients in clinical trials. And I know Hank Meyer has been incredible when it comes to advancing those outcome measures for pulmonary outcomes so that we can expand into that population.
Right, yeah. If we don't develop those outcome measures, we really can't then take those investigational products and test them and see if they're working to get to an approved therapy. And that's a challenge we always face.
Yeah.
And I think we are good to go and send it back to Dr. Meyer.
Yes, my apologies for the technical challenges here. So as an introduction to management, I'd like to talk about how an acute illness develops. It first requires an infection, whether it's a virus or a bacteria. Then what has to happen is that infection needs to contact the respiratory epithelium. And then at that point, which the respiratory epithelium is the lining of the respiratory tract from the nose down all the way into the lungs. Then what happens from there is that there's an immune response and an inflammatory reaction, and that's the body's response to the infection. And that involves antibody production, and antibodies are important in being able to recognize foreign infectious material and then mark them for destruction. And part of that process involves the production of something called cytokines, which cause inflammation. And a lot of the general inflammation and fever that you get during an infection. And then there's a rush of immune system cells that go to the point of infection, such as in the lungs, and then into the airways to actually fight the infection. That process then produces secretions both from the inflammation and then from the migration of white blood cells, which then produces purulent or pus-thick secretions. Next slide, please. So the most critical response in response to an infection is cough. The issue, however, is if you have, go to the next slide, please, a neuromuscular condition, then that requires coughing supplemented with something like the cough assist or mechanical insufflation exsufflation, as Dr. Schell had introduced. Next slide, please. So the response to an acute illness involves clearing the upper airway with coughing and suctioning. And then in addition to clearing the upper airway, you're protecting the lower airway to make sure that whatever material may be in the upper airway stays there or is removed and does not go down into the lower airway. Then the final part of coughing and suctioning is to clear whatever is either produced in the lower airways or migrates down into the lower airways from above. The issue that we sometimes get into is how often can you use a cough assist or an airway clearance device? And the simple answer is as much as you need to. When we have to place orders for nurses, we'll often place an order that has an additional as needed order for something perhaps as frequently as every hour or so. The other thing that's required during an acute illness is rest. Whether you have a neuromuscular condition or not, having an infection is very energy draining. And so for our patients with neuromuscular disease, respiratory rest, which sometimes involves increasing support with non-invasive ventilation. Next slide, please. So the next thing I'd like to talk about is at what point do you need to change from home management to seeking acute care in the emergency department? And the simple answer is when what you need to do at home can't be done safely. And that involves both the frequency of treatments and the intensity. And the other thing that would prompt me to recommend a trip to the emergency room is when you begin to have a persistent need for supplemental oxygen. Many of our patients will be home with oxygen to use for very brief periods of time because, of course, it's not something that we like to use chronically because that often covers over a more important problem that needs to be managed. If you require supplemental oxygen, that typically means that you have secretions in the lower airway that need to be removed and may be the beginning of more aggressive deterioration. And so if you start to need supplemental oxygen, it's absolutely important to seek more involved medical care in the hospital. Next slide, please. So the question then is how to go to the emergency room. And the answer is prepared. And what does that mean? Let's go to the next slide. need to assume when you go to the emergency room that you're going to be the expert, even though you may not be a doctor or a nurse. What I recommend is notifying the pulmonologist or respiratory physician involved in your child's care, and then go with all of the equipment that you need for routine management, whether that's a cough assist, suction equipment, the ventilator, or any other medications that may be used. If you have medical history folder or prior chest x-rays from when your child is well, that can also be very helpful. And that will obviously be useful in helping to determine if a change that may, or I'm sorry, a concerning finding that may be present on history or physical exam or on a chest x-ray is new and needs to be managed. The other thing that may be helpful for some is bringing a standard of care document, like many of which have been, like what's been produced through Parent Project for Muscular Dystrophy and separately by Dr. Berkrant, simply because you may have people that just aren't appropriately familiar with neuromuscular management. Next slide, please. So that involves being willing to be an advocate and then educate the people in the emergency room. That sometimes involves going a step further and being a very strong advocate for your child, which may, to some, make you, in their words, a difficult parent, which I think is a good parent. You need to be a strong advocate. Next slide, please. So, what are the potential barriers that you may face in the emergency room? The first thing is you may confront people that are inexperienced in managing patients with neuromuscular disease. When that happens, it's important to do your best to educate them and try and work as a team. The other thing is having physicians, nurses, or respiratory therapists that are unwilling to listen to your child's expert, which is you as a parent. And when that happens, the best recommendation I can give is to be persistent and consistent in what you're advocating for. One of the new challenges that we face now with the COVID pandemic is that hospitals try very hard to limit what we call aerosol generating procedures, two of which are central to neuromuscular care, that being airway clearance with a device like the cough assist, or IPV, intrapulmonary percussive ventilation, or nebulized medication, and ventilation, whether it's invasive ventilation by a tracheostomy tube or the more common non-invasive nasal ventilation or oral mouthpiece ventilation. Next slide, please. The question that's often asked as the first question upon arrival is, at least rhetorically, is this somebody that has COVID or SARS-CoV-2? That usually then leads to testing. And until negative, what often will happen is that the patient will need to be in a special isolation unit or room, and the people that take care of the child will be expected to wear extensive protective equipment. That will continue until the COVID status is determined to be negative, or if the patient happens to be positive, then that level of isolation will need to continue further on. Next slide, please. So, in my hospital, there was an initial attempt to try and limit aerosol generating procedures. And the guidance initially was to decrease the frequency of use of airway clearance and non-invasive ventilation, or to turn down the pressure that's being used. I jumped up and down and stamped my feet, and with working with our infection control people, came up with an alternate approach. Next slide, please. And that is instead to not do anything to decrease the frequency. I'm sorry, can you go to the next slide, please? Not to decrease the frequency or pressure used for airway clearance, but instead to take the aggressive approach looking from the perspective of trying to limit the severity and duration of the illness. And that requires early and aggressive management of both the airway clearance and ventilatory support, and then switch to trying to limit the to limit exhalation from the ventilatory equipment by switching from humidification to a heat moisture exchanger, and then switching to inhaler treatment when appropriate. And when there's need for invasive ventilation, to try and use what they call a closed circuit, so exhalation is not back into the ambient air in the room. Next slide, please. So the treatment strategy involves two things: oxygenation and ventilation. And they're obviously integrated, but often are looked at differently in an acute situation. To manage oxygenation, the first approach is to try and clear the airways using devices that should be quite familiar to many of you, such as the cough assist and chest physiotherapy. The one thing that we don't want to have is our patients on oxygen without appropriate respiratory support, or without clear guidance, clear information that says that the carbon dioxide is normal. What usually happens is that, coincident with using oxygen support, if that's necessary, patients will be on mechanical ventilation because of increased breathing muscle weakness. And in that situation, we will use supplemental oxygen if needed. Next slide, please. So if we shift from doing the acute initial management into the maintenance phase, what we want to do is support the body while it's fighting the infection. And that involves continuing aggressive airway clearance, even if the oxygenation varies somewhat. When the oxygenation varies, in other words, if you have low oxygen acutely, that often is an indication of mobilizing secretions, and it's important to continue airway clearance through that. The next thing is we need to continue with ventilation to keep the carbon dioxide normal, and sometimes we need to have, add additional medications such as antibiotics or antiviral medications, which are sometimes used for COVID, and sometimes medicines to help to thin the mucus to make it easier to clear, and occasionally some additional devices such as the vest, which Dr. Schell had mentioned, and interpulmonary percussive ventilation. Next slide, please. Invasive ventilation by an endotracheal tube is often used, or is used, when non-invasive nasal or oral or mouthpiece ventilation is unable to provide the support needed for effective ventilation, or we're not able to maintain an adequate interface to get the air into the patient. Having an endotracheal tube, or a tube through the mouth, is not equivalent to having a tracheostomy tube and shouldn't be considered as a step in that direction in the acute setting. Extubation, on the other side, when the infection has resolved or is resolving, is every bit as important. And the recommendation is to not look to removing the breathing tube until until the supplemental oxygen requirement has resolved, which would indicate that you've done an effective job of clearing the airways. After the tube is removed, it's very important to spend time clearing the central airways of excess mucus, and continuing with that until the patient is on room air, needing no supplemental oxygen. Next slide, please. The next step is de-escalation, and that involves returning to baseline, or at least to a level where care can be managed at home. And that involves a discussion between the treatment team and the family. What I think is very important is to follow oxygen and not accept needing extra oxygen in exchange for decreasing airway clearance or respiratory support. The other thing to keep in mind is that any significant infection, such as coronavirus or any other respiratory virus, can cause inflammation that may take some extra time to recover. And so sometimes we will set the goal of not returning the patient's respiratory support to the exact settings they were before they got sick, but to a level where the family can manage effectively at home. And that, again, is part of a discussion to have with the treatment team. Next slide. So, to conclude, back to baseline is really what we're trying to look for, and that involves returning sometimes to a new normal. It's absolutely critical at the early stage of an acute illness to be aggressive in airway management, and that will do a lot to limit the severity and duration of an illness and improves the likelihood of a successful outcome. And with that, I'll turn back to Ryan and Eric. Thank you for your attention.
Thank you, Dr. Meyer. We really appreciate your insight on how to prepare for those kind of emergency situations. As a reminder to our families at home, we do have emergency care cards. You can be found under the support and care materials on the care tab at parentprojectmd.org. And with that, we are going to move on to our next speaker, Dr. Lisa Wolf from Northwestern Memorial Hospital, and she is going to discuss the lessons that we have learned from COVID-19. So, Dr. Wolf. Hi, Dr. Wolf. I think you're muted right now.
Sorry.
Oh, there you go. Yep, perfect. Thank you.
So can I ask you to go to my first slide? Okay, so basically what I'm talking about here is just that we've made a top 10 list of lessons learned from COVID-19 for our muscular dystrophy patients, and we're going to go through some of that top 10 list with you today. Next slide. So first, the first question is, what should we ask our caregivers to do to be safe in our homes? Many of us have nurses and paid caregivers that are coming into the home that have been out and about during the day. It's important that we ask them to use hand sanitizer when they first hit the door and to keep liquid soap available with paper towels that can be disposed of to use frequently during the day. You should ask your caregivers to do surgical masks with universal masking, meaning that they're worn for the whole day. You should ask your caregivers to avoid travel outside the area, and that if they are sick, they should avoid coming to work. Now, the other thing is, if they're exposed to someone who's sick, they should also avoid coming in. And that can be sort of the more challenging part. Next slide. Number 2, what if I can't find protective gear for my caregivers? So this is a common problem because often our nurses and caregivers don't work for hospitals or large agencies. And as such, they don't have ready access to masks and gloves. What we would encourage you to do is to extend the life of your masks, especially by providing your caregivers with paper bags that have their name on it, where they can put their surgical mask in the bag in between shifts and continue to use it until it is soiled or broken or wet, so that we can extend the life of the mask. If you're having trouble finding masks, talk to your school district, especially the school nurse, who may have access to masks and other protective equipment. If you have friends that are crafty and can sew, ask them to make you a mask. People are really excited to help and they want to. We just have to give them some direction. If you have in your community a community-based living organization, then I would reach out to them because they might have some access to PPE, and they may do it in a way that is allowing it to be available at a much cheaper price or even for free. Lastly, I would ask your caregivers to do what we do in the hospital, which is consider wearing scrubs when they're at the house for the shift and change back into their regular street clothes when they're ready to go home. Next slide. Okay, the third issue, what can I do to make it safer to go to school? So this has been a very, very hot topic, and certainly every school district has its own rules and its own plans. So these are just some general things that can help if your school is opening and you plan to go to school. First of all, limit exposure to as few people as possible, and avoid public transportation if you can. If you absolutely have to go to school on a bus, try to stay with one bus aide and one aide in the classroom. The more people you're exposed to, the more risk you put yourself and your family to. Try to stay in the same classroom. So instead of moving from classroom to classroom throughout the day, see if the teachers can come to you. The less that you're exposed to different environments, the better off you're going to be. We want our students to eat in very open areas because they'll have their mask off at that time. Often a communal cafeteria is not the right place for that. And as such, a student may need to eat in their classroom. Don't be afraid to stay home. If you feel unsafe at school, if you feel that your school district can't meet your needs, remember that our patients with Duchenne have a significant increased risk for infection, and you can discuss with your school district to make it a part of your IEP to allow you to stay at home. Please avoid sharing textbooks or other supplies. Anything that you use should be for you only. And lastly, don't forget that our teachers frequently have to provide their own supplies in the classroom. So if you can, donate cleaning supplies and hand sanitizers. They're going to be in need. Next slide. Question number 4, should we get a flu or pneumonia shot? And the answer is yes. We've already spoken today about Dr. Bernkrant's wonderful paper in Lancet, helping us to understand what the standards of care are for Duchenne muscular dystrophy. Notice that right up front is the recommendation that there's an immunization with pneumococcal vaccine, and that would be every 5 years, and a yearly vaccination for influenza. There is a fear that we would have a second or additional wave of coronavirus that would coincide with our seasonal episodes of influenza, and that would significantly impact the morbidity and the mortality that we see. And so it's going to be more important this year than any other year to make sure you get your influenza vaccine. Next slide. Number 5, should I continue taking steroids? Well, our patients with muscular dystrophy are taking steroids, especially for Duchenne muscular dystrophy, because it positively impacts inflammation in muscle, helping to keep that muscle strong, both heart and lung muscle. However, the steroids have side effects, namely diabetes, which increases your risk of getting infections, as well the obesity that can be associated with the use of steroids increases risk of complications with the COVID virus, increasing risk for need for ICU, intubation, and complications after disease. So number 2, what are the concerns for stopping it? Well, they're significant. Stopping your steroids cold turkey could cause significant weakness, and weak muscles could reduce your cough strength. Weak muscles include weak cardiac muscle, which could significantly increase your body's ability to maintain normal blood pressure, heart rate, etc. So are there adjustments that we can use that will help us to adequately care for our bodies and still use our steroids? There are. If you get sick, talk to your doctor. They may need to actually increase your dose. We call this stress dosing of the steroids to compensate for the needs in acute infection. Also, they may switch your steroids to dexamethasone, which is specifically used during the COVID infection. So the bottom line is, final answer, don't stop your steroids. Next slide. So If we look at our other medications, that one that is most commonly used for our boys with Duchenne muscular dystrophy would be ACE, or angiotensin converting enzyme inhibitors. These drugs have been shown to improve the cardiac outcomes in boys with Duchenne muscular dystrophy by helping to reduce remodeling or loss of cardiac muscle. However, there is a theoretic problem, which is illustrated in the cartoon on this slide, that because the COVID virus requires the ACE receptor in order to go into cells and infect them, and by taking an ACE inhibitor, you increase the number of ACE2 receptors on the cell, there's a theoretic concern that taking an ACE inhibitor may be problematic. However, we also know that ACE inhibitors protect the lungs from damage, and they also help to protect the heart. And because of that, there is a strong recommendation from the American Heart Association. Final answer, do not stop your ACE inhibitor. Next slide. Number 7, will I be able to get my respiratory supplies? Well, for those of you who have gone to the grocery store and seen empty shelves, although they're much better now around here anyway, the respiratory therapy department at your home care company is going through the same thing. They may not have as many parts for their PAP machines or nebulizers, and they may not have as many medications, especially those albuterol inhalers. What can you do? Well, request your refills or resupplies early. The earlier you do that, the more you'll be able to compensate for delays in our system. Use your parts for longer. If your mask is still working, try to get it to work a little bit longer so that you will be able to preserve your current supply if we have ongoing shortages. And don't get frustrated. Your home care company is likely doing the best that they can. Talk to your physician. They may have some extra samples or hospital supplies that can help you out, but know that you're not the only person going through this, and together we'll come up with solutions to make sure everyone gets their supplies. Next slide. What if I need a home ventilator? So a home ventilator, sorry about that, a home ventilator is often used as a way to provide bilevel or BiPAP. It's a device that also can be used in the hospital in order to provide support for someone with a COVID infection. And because of that, many of our home ventilators have been reassigned to the hospital and may not be available through your durable medical equipment provider. So some of the things that we've done to help compensate for this reduction in available resources, we may temporarily have to change the brand or version of the machine that we give you. You might need to use a smaller PAP machine and then buy an external battery yourself. And you may need to go with a single device in the home instead of 2. All of these are temporary fixes, and eventually, as devices become available and on the shelf again, we can get patients a more specific or more appropriate device as needed. Next slide. If I go to the hospital, will I be denied care, especially with non-invasive ventilation or airway clearance? So Hank just went through with us the challenges he has. And Hank, what I really want is I want to be there the day when I can see you jumping up and down, because I would have paid good money to see that. Anyway, those respiratory devices can generate aerosolized particles. And as we talked about before, can be hesitant for hospitals to give you your equipment. What you need to find is a champion. If you have a champion at your hospital, they will be able to make sure that policies are appropriate for our patients who have muscular dystrophy. Those enlightened policies will be there, but as an independent advocate for your patient, you by yourself may not be able to get there. So find your champion. Bring your own equipment for home and make sure that they know that oxygen by itself could be dangerous for you. So, next slide. This is the last of our top 10. What if I go to the hospital and I'm denied critical care? Well, there's a lot in the news about that right now. There are definitely discussions about patients with disabilities who have been denied care in areas that are being overwhelmed and in systems that are out of ICU beds. And it would be foolish of me to talk to you today and not at least address the question. So, theoretically, if our systems are overwhelmed, you could be at risk of not being offered care, but bringing your own equipment will make a huge difference. Make sure that you bring a caregiver with you who knows how to run your equipment. But in the end, the most important thing you have is your champion, the person at your hospital that you can identify who will be there to advocate for you. The most important thing you can do today, find your champion, cheer for your champion, and let them know you're there for them. So I am out of time now, and that is the end of my slides.
Thank you, Dr. Wolf. That was great. We've had so many questions come in around these areas with COVID-19 ongoing, and so I think that helped a lot of families. So now we are going to take this to the full panel and have a Q&A. So again, if you have any questions, please submit them in that box below so we can address them during this session. And with that, I am going to hand it over to Rachel Schrader. So Rachel, it's all yours.
Thanks, Eric. I think that was such a great conversation about pulmonary management and how to avoid emergencies and then what to do when we face those emergencies. So I look forward to a great question and answer session with our different speakers. So I think we'll start off with you, Dr. Meyer. One of the questions that came in is regarding people who have Duchenne and are steroid dependent. So in this particular case, let's assume that the children are younger, maybe grade school age. How concerned should parents be about COVID-19 and their respiratory health of their sons with Duchenne?
That's a very good question. And the answer I would give is essentially the same concern that you would have during the height of flu season any year in the past. The difference with coronavirus is that there's very little immunity within the community, and so it's been spreading quite dramatically. At least our pediatric experience has been relatively good in that our patients, we haven't had thankfully experience with any neuromuscular patients here in the hospital, but our pediatric patients typically do quite well and recover and go home. And so while I would be concerned about any neuromuscular patient being exposed to a respiratory virus, I haven't been more concerned about coronavirus per se as a specific pathogen. I don't know if Lisa or Richard have additional experience. Of course, Lisa has a lot more experience with the adult population than I do.
Yeah, I would definitely say that our adults are much greater risk than the children are for a couple of reasons. One of which is that their cardiac function is already impaired, which makes it a great deal more difficult to handle any infection should they get it.
I agree with both of you. I also would mirror the fact that we here in Columbus have not had any Duchenne patients or neuromuscular patients admitted with COVID from a pediatric age standpoint. So, but, I've been also telling parents that because of these risks that, that we can't really quantify, except to say that they are at greater risk.
Wonderful. Thank you so much. The next question is for Dr. Schell. So in addition to using a cough assist device to move fluids, I know it can be used for pulmonary exercise as well. Could you tell us a little bit about the best protocol for lung recruitment or breath stacking?
So that's a great question. And I think it kind of hits home to my point that the use of the cough assist or, any type of manual insufflation, exsufflation is I personally think it's really great physiotherapy for the lungs or physical therapy for the lung muscles, especially if you are not as mobile, if you are in a wheelchair for most of your day or sitting down for most of the day. Using a cough assist to help take those deep breaths and open up those tiny places in the bottom of our lungs and help clear those, I think, is important. I usually recommend, to find a setting on the cough assist or whatever device that you have to help that augmented cough at home that's comfortable for you. And some people tolerate settings of 20 and some people tolerate much higher than that. And finding a comfortable setting that's both an inspiratory setting and expiratory setting, they don't have to be exactly the same. I ask my patients to do it twice a day as a regular thing, like brushing your teeth, like I said. I know that not everybody does that twice a day. A lot of our patients will say, well, they didn't really use it, or we fell off the bandwagon using it every day. But we use it when they're sick, but they haven't been sick for the past 6 months. So, my gut tells me that the more people use it, the more stable their pulmonary function may be, though we haven't really been able to prove that.
Wonderful. Thank you so much. This next question is for Dr. Wolf. So given the high levels of COVID-19 in some states and cities, do you think it's safe to do a sleep study to titrate my son's BiPAP machine? I think this is maybe something that we think about a lot with maybe some of our adult or, older teen patients. I'd love to hear your perspective.
So the first thing is each sleep lab is handling this situation differently. There's no universal here. What we have seen is that many labs are doing COVID testing before doing a PAP titration, and that they're requiring their techs to wear full PPE, which includes an N95 mask and a face shield. So for instance, if you were coming to our lab at Northwestern, we would do a COVID test within 72 hours of your sleep study. You would come in by yourself. You wouldn't wait in a waiting room. You'd go straight to your individual room, and your technologist would only enter that room if they were wearing full N95 face shield, gown, and gloves. Given that scenario, yes, in fact, you're much safer going to your sleep study than you are the grocery store. And so I think it's important to ask what the lab's plan is to handle the risk for COVID. And I think that you'll find that labs that are affiliated with hospitals rather than standalone labs are doing a much better job. And it's only because access to COVID testing and access to PPE has been a huge struggle for the private labs, where the hospital labs have better access.
That's a really helpful explanation, Dr. Wolf. In that same vein, Dr. Meyer, what would you say for a family who has been experiencing perhaps having their PFT testing canceled? Is this something that they should advocate for having regularly, even during the pandemic?
I will tell you from our own experience that we limited pulmonary function testing at the height of the pandemic to patients where the data was absolutely critical for clinical management. And that pretty much meant just patients on oncology protocols. Over the last couple of, month and a half or so, since we've begun to see our patients in the office, we've been opening up testing to whatever patients are here for clinical visits. I personally don't think that a single pulmonary function test in a patient with Duchenne muscular dystrophy, missing it is all that critical in the overall management. If there is something that's felt to be critical, then and it can't wait, then I think it would be fine to arrange for a pulmonary function visit in the hospital, but I wouldn't lose any sleep over missing the occasional pulmonary function test.
Wonderful, thank you. All right, so Dr. Schell, what are your thoughts about using mouthpiece ventilation? Isn't it better to use mouthpiece non-invasive ventilation with a straw during the day and maybe a mask at night instead of getting a tracheostomy that would potentially increase your risk for infection?
So that's a good question as well. I wouldn't say one is better than the other. I've always told families that this is a choice. I mean, in fact, for instance, I just had a young man who, he didn't share with us that he was using his BiPAP 24/7 until this most recent visit. And he was happy doing that. But when we talked about it, we talked about the choices. And he decided that he would like to try a SIP valve. And I didn't really go into it in detail in my talk, but a SIP ventilation is a straw basically that's attached to your ventilator that, when you need to take an extra breath during a conversation, you can turn your head, your straw's right there, and you sip off the vent, and it'll give you that breath that you need. So it is something that many patients will choose, and it can be very comfortable. It's somewhat cumbersome sometimes depending on your home care company to set it up, but once it's set up, most patients really like it. But I also have patients that choose that, something, I'm using SIP ventilation all day and I'm using BiPAP for more than 18 hours during the day and night, and it would be more comfortable to me to have a tracheostomy tube where the ventilator can just breathe for me all the time, and I can do other things. So it's really a patient choice more than anything.
Great. Thanks so much, Dr. Schell. So Dr. Wolf, you mentioned a little bit about access to different equipment needs potentially during hospitalization in regard to the pandemic. Are there any laws that would prevent Duchenne patients who need care from being turned away at the hospital that you're aware of?
So what you're saying is, could a hospital okay? That was sort of a double negative question, but let me just try to answer it. The bottom line is that the hospital is absolutely obligated to treat you. If there was, God forbid, a time in which our systems were completely overwhelmed, and for those of you that live in environments where there are no ICU beds, where they're completely used up, and where there may not be access to a mechanical ventilator, there are laws that allow a hospital to say, we're going to potentially decide who gets that vent and who doesn't. I mean, they're going to have to if they have limited resources. There are no laws that say you have to treat a patient with Duchenne muscular dystrophy, including a mechanical ventilator. They have to treat you, but that doesn't mean that if they were in a situation in which resources were limited, that they would have to give a limited resource to this one or that one. It's going to have to be assigned to someone and not given to someone else. What we are very hopeful for is that if we can stop the spread of the virus by having people do good hand washing, isolation, mask use, etc., that we allow our healthcare systems to be in a position where we can care for everyone, where we're not overwhelmed, where we have enough beds, we have enough ventilators, so that, God forbid, we will never need to be in that position.
Wonderful, thank you. And a follow-on question that maybe Dr. Meyer, you can start us off, and I think this will probably be our last question. How does a family find that champion in their own hospital? What if they can't find a champion? Where can they go for help?
Oh boy, that's a good one. I don't know that I have a great answer other than to offer the expertise that the three of us here on the call have, and the willingness for us and others to be willing to talk to local physicians. I've had the opportunity to talk to very engaged, intelligent, and active physicians that just need a little bit of help figuring out what to do. Having said that, I think the most useful approach is being the expert and hopefully finding somebody that's willing not so much to take orders from you, but willing to work with you and understand how to take care of your child. Having said that, it can be difficult because physicians and nurses are also highly educated and know medicine well, while they may not know your child well. And it's important to try and find somebody that's willing to be educated. The other thing that I just wanted to address very briefly and align with Lisa's last question is that a lot of, I actually, before I was on this meeting, had a call from a mother with another neuromuscular condition where she was concerned about the perception that her hospital in New York, where the pandemic hit quite violently a couple of months ago, the perspective that the hospital would have based on how the condition her child had was phrased, be it severe, mild, or moderate. And so I think some of the response to a hospital may be in the documents that you come with saying, my child has Duchenne muscular dystrophy and is exceptionally well controlled at baseline with the support that Dr. Schell has given him or Dr. Wolf has given him, with the opportunity then to talk to a treating physician.
Great. Thanks so much, Dr. Meyer. I wish we had more time to go through some more questions, but, this has been a really great conversation. And I'll also just, give a quick plug. We've got lots of information on our website about pulmonary care and interventions, and a care team, including myself and some other great folks on the team. So if you have questions, we'd love to chat with you after the fact as well. And with that, I will turn it back to Eric and Ryan. Thanks so much, everyone.
Thank you, Rachel, and thank you to all of our panelists for that great discussion. We're going to take about a 45-minute break so everyone can go grab some dinner. When we come back, we are going to have our final quiz, and we're going to get into our gene therapy session. So we will see everyone back here at about 7:10 Eastern.
Welcome back, everybody. We are about to start the highly anticipated gene therapy session, but of course, I'm going to make you do some polling. Polling, once again, is for all of our families. You can access the polling by pollev.com/PPMD. Let's get the first question up. This question is, do you agree or disagree with the following statement? When I have questions about the science of gene therapy, I have enough educational resources available to me to answer those questions. Do you strongly disagree? Do you disagree? Neither agree nor disagree, agree, or strongly agree. Give a second for people to answer these questions.
And this information is really helpful for us as an organization, as well as the sponsors that we're going to be speaking to in a little bit, just to find out what we need to work on and better disseminate information if it needed. But we are seeing a lot of agree and strongly agree, and it's great to see.
And these polls are anonymous, so do not feel intimidated. We want you to be honest. It's important that you answer and give us the best possible feedback so it can inform the work we do.
But it is a complex topic, so we know that we need to have a lot of things there available for you. There can be a lot of questions about this. As we've seen already, we have a lot coming in for this session, so we're excited for that.
Okay. Got a few strongly disagrees. Okay, with that, I think we should move to the next question. Next question, in your opinion, do you feel the following sentence is true or false? If one gene therapy doesn't work, my child can always try another gene therapy. And we're going to talk about some of this today.
Yeah, we're definitely going to address this topic, talk about some of the aspects of that. And this is something that we have asked at previous conferences. And it's kind of, along with that education question, a bit of a benchmark, I think, for us in terms of, how we're doing with the community, in terms of making sure there's, educational tools out there.
Yes, absolutely. This really informs the work we're all doing. There's so many in our drug development ecosystem working on gene therapies. So your voice, your information informs what we do. So it's so important to participate. That's why when we give you polls or surveys, they don't just go nowhere. We actually use that to inform the work everybody is doing.
And it's very early days still with these therapies, so uncertainty is to be expected.
Yeah.
So not surprised to see a number of unsure.
That's folks are unsure. Yeah. Okay, we're going to move on to the next question. So this one's going to take a little bit more time to answer. Imagine a scenario which your child has the potential to join a gene therapy trial. What would you need to know in order to help make a confident decision to join? You can choose your top two here. So you're able to choose two choices. I'm sure many of these are relevant.
Right. So what are the top two? So, information coming from your doctor, the details about the gene therapy itself, what kind of data is out there, which, is always important. We want to know, are these safe? Are they working well? Which is, something that's important to everyone. And it looks like, that data from human studies is kind of winning the, what's most important over something like the preclinical data.
Mm-hmm. Mm-hmm. And we also, we're very used to shared decision making. Recommendations from your doctor matters when it comes to choosing a trial. We see this in our other polling and our other data, so not surprised to see that high in the list.
Especially for something like gene therapy, there's a lot of considerations there, so that is something that we do always recommend, have those conversations with your whole care team to talk about what's available to you and your family, and what that might mean to participate in some of those trials.
And these forms will hopefully allow you to ask those questions. You can write them down during these conversations you're going to hear today. Okay, we're going to move to the next question. Okay, knowing what you know today, how comfortable would you, your child, be joining a gene therapy clinical trial? Again, all this is anonymous. We just want your honest answers.
And, we've done some similar work kind of along this question before with some of our patient preference work around gene therapy, and risks associated with that.
Yeah, absolutely. And preferences change over time in terms of as new knowledge comes about, as the field advances. I think everyone's going to be evaluating their benefit risk and choices when it comes to clinical trials, and it's important to keep the community informed so they can have those conversations with their doctors.
And again, this may be influenced by where you or your child is in the age or stage of the disease. So all factors. And again, we're no judgment on whether you're super comfortable or very uncomfortable. That's a decision for you and your family.
Yes. Okay, I think we can go to the last question, and this is our pop quiz. In what year did Apple Computers release the first iPod? I think I had one of the first iPods. I'm pretty sure. No, it was the size of my computer, I think I remember.
No, I did not have one of the ones with the click wheels, which I believe were those first generations.
Well, you just called me old. That's okay, though. See, the answers are still trickling in.
This was definitely one that I got wrong earlier today.
Okay, so the answer actually is 2001. So whoever answered it first will reveal the prize later on. We have a packed sort of session ahead of us. So we may have to reveal prize winners tomorrow, and that's okay, but I'm going to kick it to Eric now, who's going to start us off with this session.
Thank you, Ryan. So tonight we're going to be talking about gene therapy. As part of that, to introduce us to this topic, because there is some complexity here, we are going to be joined by Dr. Tim Kripe from Nationwide Children's Hospital, and he's going to give us kind of an introduction and an overview of gene therapy before we are joined by the rest of our sponsors. And just so you know who else you're going to be hearing from tonight, our panelists are going to include Dr. Louise Rodino-Klapek from Sarepta Therapeutics. We have Dr. Beth Belluccio from Pfizer. We have Dr. Fulvio Mavilio from Audentes Therapeutics, and Dr. Joel Schneider from Solid Biosciences. As a reminder, if you have questions, please submit them to the comment box below. We've already had a ton of questions come in, so I think we're going to have a great discussion after this. But to begin, we are going to start with an introduction by Dr. Kripe. So, Dr. Kripe, I'm going to hand it off to you.
Thank you, Eric. It's a pleasure to be here and be participating in this fantastic panel. I guess it's my daunting task to give a brief introduction to gene therapy, which is a large field, a complex field, a lot of nuances, and we're learning something about it every day, it seems. So I'll just give an overview to hopefully get everyone up to the same page, and maybe it'll help some of you understand what comes after this talk better in terms of the other presentations. So the first slide really shows the promise of gene therapy, and that is that you could correct a diseased cell by replacing or changing or altering expression of a protein. So a diseased cell that's missing a functioning protein in its simplest iteration can be replaced with gene therapy and result in a healthy cell. So that's really the ultimate promise. And of course, that sounds easy, but is very challenging in a whole organism and with lots of cells and different kinds of diseases to attend to. But what exactly is gene therapy? On the next slide, you can see that I've broken the definition of gene therapy down into very simple subsets. That is the use of some sort of a nucleic acid, DNA, RNA, sometimes hybrids of those two, or a mimic of those, chemical mimic, to either modify, replace, or add genes. So modifying gene expression or splicing. I showed an example of that with Duchenne muscular dystrophy with eteplirsen. Or to replace a defective gene, as there's two FDA-approved versions of replacing a defective gene, Luxturna and Zolgensma. Or adding a new gene, and that's what's under trial right now with Duchenne, but there is an FDA-approved version of adding a new gene, and that's in the cancer field. It's something called Kymriah. Now, that's not happening in the body. It's where cells are removed from the body and they're modified, new genes added, and then they're given back to the body. So it's a little bit different paradigm. But gene therapy really encompasses all of these approaches. So on the next slide, it shows a lot of different methods of gene therapy. Many people use viruses because viruses are small particles that have evolved over thousands or millions of years to basically transfer nucleic acid into a cell. That's what they do. They're shuttles. They bring it into the cell and then they affect the cell. And there's a lot of different kinds. There's thousands and thousands of different types of viruses, many of which are under study. as gene therapy vectors, which is a term we use to refer to the shuttle, the particle that brings in a nucleic acid. And so even HIV has been used for that. And in fact, that's the one that's used for that cancer application. But among those lists is a parvovirus called adeno-associated virus, or AAV, which is the most commonly used now, and particularly in the Duchenne field. And AAV, despite its name, is not an adenovirus. So the Oxford, for example, the Oxford COVID vaccine is using an animal adenovirus, but it's very different from an adeno-associated virus. They're completely different types of viruses. So if you got the COVID vaccine, you wouldn't become immune to an AAV. They're very different. But also, many people are looking at other ways to transfer nucleic acid into a cell, such as non-virus mediated. And these have been around for many years as well. They tend not to be as efficient, but technology is improving all the time. And so there are many applications that are being tested, including packaging DNA into various types of particles, like lipid particles, liposomes, or condensing them onto other kinds of substrates, or even injecting them by themselves, we call naked DNA. And then there's been studies done with a gene gun where DNA or RNA are coated onto beads and shot into the tissues, believe it or not. So the next slide will describe, so that's just some of the various ways, but I'm going to focus the rest of the talk really on the use of AAV, since that's most common with the muscular dystrophies. And this slide gives you some basic principles about how the vector, the virus, gets into the cell. That does so by binding a receptor on the outside of the cell surface. And so that receptor has to match the virus, or it's not going to get in there. So I kind of refer to that receptor as the front door lock. And the virus has got to have the right key. And once it gets into the cell, it's got to traverse through the cytoplasm and enter the nucleus, and then deposit its nucleic acid. If it's a DNA, for most viruses, some actually work in the cytoplasm, but AAV works in the nucleus. And in the nucleus, the DNA is then begins to undergo its biology. That is, it can be turned on in terms of gene expression, and it can undergo replication or copying of itself to make more viruses. In the case of most gene therapy applications, they're defective. They're not going to make more copies of themselves. They're just in there to get that gene into the nucleus, to turn it on and to affect its function. And the promoter, which is a term we use for the regulatory sequence on the DNA, that's not the actual coding sequence for a protein, but it's basically the turn-on switch, determines what cells it's expressed in and how much is expressed in that cell. So I kind of refer to the promoter as a speaker volume. So the choice of the virus serotype, or the different type of virus with the key, will determine which tissues it goes to, and the promoter will determine how loud or how much that gene is expressed in those tissues. Now, on the next slide, it shows that some different kinds of what we call serotypes, or those different keys, will go to different target tissues in different ways and in different amounts. So different serotypes tend to infect different kinds of tissues, but this is by no means an exhaustive table, and it just gives you a flavor that some tend to go to muscle more than others. Most of them do go to liver, because there are particulates, and liver is a big filter for the bloodstream. And they may also go to the spleen or the kidney for the same reason, sort of a non-specific uptake. But many of them will bind that lock and key in different kinds of organs. So some of these serotypes are better for nerve tissue, for example. But very few examples only go to a single tissue. I think folks are trying to engineer those now to be very specific for a different tissue. They may preferentially go to certain tissues, but most end up more than one place in the body. On the next slide, it sort of illustrates one of the principles that scientists have undertaken to try to not just rely on what nature's produced, but to actually make our own different variants of this capsid shell, this vector, this shuttle, because we can mix and match different amino acids, different proteins, and make different capsids and result in a variety of different kinds of capsid shells, and even engineer different mutations in there, and select for different variants that might preferentially favor going to one tissue or another, to try to increase that selectivity for a different tissue type. So if we look on the next slide in the virus itself, The wild type AAV, adeno-associated virus, meaning the wild type, meaning the kind that's found naturally, has two major genes. It's got inverted terminal repeats, the ITRs, that help it package into that capsid. And then it's got two major coding genes, and actually there's 6 or so other genes, proteins that come out of these by different splicing and different promoters. But what we do in the genetic engineering world is to remove those genes. So it's no longer a replicating virus on its own. We actually provide the function of those genes that form the capsid on another plasmid or in another system, but we're able to then, what we call, gut the virus and replace the normal genes of the virus with the genes we want to get into a cell. So you can see on the bottom, the recombinant version shows the transgene in green there, which is the gene we want expressed in a cell, and then the promoter, which is that speaker volume to turn on that transgene. And there's usually some other regulatory elements, and here you can see a polyadenylation signal to get that gene to be expressed correctly. And the downside of AAV vectors is their very limited capacity, about 4.7 kilobases, 4,700 bases. Here it says 5,000. So around that area in terms of the amount you can package in that capsid shell. There are other viruses that can package more, but they also have disadvantages or advantages. Every virus has pros and cons. So this most common AAV has that limitation, which can come into play when one's considering which genes to turn on or off. Now, on the next slide, you can see one of the challenges, these are the different promoters, just examples that one can use: strong promoters that are active across many cell types, or tissue selective promoters that might usually be less strong, and they're not usually 100% specific, although we're getting better all the time at making promoters be very selective to different cells. So there's a lot of different genes, promoters in nature that one can take to selectively express a transgene in muscle or in neuron or liver, for example. And nowadays we have combinations of different sequences or synthetic promoters to try to really tune that expression better into the cell type that we want. So on the next slide, we see some of the challenges of gene therapy, and that is because we're putting a virus in and the virus isn't natural, it can be seen as foreign, and so we can have immune reactions to the AAV. And interestingly, many of us have actually been exposed to AAV, different types of AAV naturally. AAV is a virus that doesn't cause disease. It's not associated with any disease. So there are AAVs sort of circulating around the population, and many of us get exposed to those as we age, and we can have an underlying immune response to those that we don't even know about. So this immune response can pre-exist to a given AAV serotype. Now, the immune response is usually separated into two major areas, the humoral and the cellular. The humoral means the antibodies that are circulating in the bloodstream. And those antibodies can bind to the virus capsid immediately when it's being infused and prevent it from getting to the tissue that we want it to get to. But if those antibodies aren't there, or if we can do something to get rid of them temporarily, which we can in some cases, then we can get the virus to the cell of interest. As you can see here, the virus goes into the cell, as I implied before, and puts its nucleic acid into the nucleus and then produces the protein. But that can result in some other kinds of immune reactions. So for example, the proteins of the capsid shell can break down and be presented on what we call HLA class 1 on the immune system, and the immune system can recognize that and generate a cellular immune response, or T cells or NK cells, against those antigens, recognizing the cell that got the virus and killing the cell altogether. So that can be a challenge. The cell that you want to express the gene can actually be killed by the immune system. That we've gotten away without that happening in many cases now, and we've shown that doesn't always happen, but that can be a problem. Now, the capsid shell we don't make more capsid shells in these cells like a replicating virus would. So once those cells, or once those capsids are broken down, and if they're cleared, then so after probably weeks or months, there may not be any more of those foreign proteins around. But the one protein that could still be there that's foreign is the transgene, depending on the situation. So if the patient is missing a certain part of the gene, and now we're providing it, that part that was missing could be seen by that individual as foreign. And so that could also generate an immune response against the protein that's encoded by the transgene, and could also result in destruction of the cell that expresses it. So those are some of the challenges with the immune response. And not only can we lose then the expression of the transgene, but we can cause damage to the organ that transgene was supposed to be expressed in. Now back to the pre-existing antibodies. This is a big problem in the field as well. So on the next slide, there's a lot of different studies out there, and this is just one I pulled just to give you a sense of that this isn't very rare. Adeno-associated virus is circulating amongst the human population, and many people have pre-existing antibodies. So, but it increases with age. This is one study that showed at age 2 to 7, the pre-existing antibodies against different AAV serotypes ranged from around 5 to 20% of people. And then as they aged, those over 8 had more in the range of 50, 60%. And that can be a challenge. So I'm just trying to raise some of the issues that many of the speakers tonight have been able to get around in various ways, and but there are also, they continue to be challenged for us. So the next slide just summarizes some of these immunologic reactions that I talked about, just to put them forefront of your mind. The immune response to new foreign protein antigens, either the virus or the transgene, in this case, a form of dystrophin, can cause organ damage or loss of transgene expression, but not in every person. Why might it happen in some and not others? It could be relating to their tissue type or their prior exposure or other factors. And then but there are various prevention strategies that are being studied: immunosuppression, plasmapheresis, and more recently, immunoglobulin cleaving drugs to circumvent this effect. And then finally, on my last slide, I just wanted to pose some of the questions that remain in gene therapy for muscular dystrophy, some of which may be starting to be answered by the presenters that you'll hear from tonight, but many of which are still lingering and will probably vex us for a while yet to come. So what are the possible risks, and can we minimize them? Risks of complement activation and platelet consumption, or putting of foreign proteins in the body, organ damage, as we've discussed, or other kinds of side effects, putting us at risk for sepsis, et cetera. What are the benefits and how durable are they? Does each type of muscle get benefit? Is it equal or not equal? And how long does that benefit last? Who is eligible for this kind of therapy in terms of age, genetic mutation, disease status, or pre-existing immunity? And what are the best strategies to enable redosing if needed? So that just summarized, I think, to me, some of the high points of gene therapy for muscular dystrophy. And so I'm passing it back to you, Eric.
Thank you, Dr. Kripe. That was an excellent introduction. I think that was going to be really helpful for everyone watching. I do want to throw one question back to you. And I think you touched on it in the presentation, but in case any of the families missed it, we have had a lot of questions coming in about the potential of a COVID-19 vaccine being delivered through a viral vector. So maybe if you can touch on that again, on the potential impact it might have on the ability to receive gene therapy, or for those who have already received gene therapy.
Mm-hmm.
Yeah, it's a great question. There are some vaccines that are being developed with adeno-associated virus. The most common COVID vaccine strategy is to take the most immunogenic or most foreign type of protein, the spike protein from the COVID virus, and express that in one of these gene therapy type platforms, so that when you get the vaccine, your body cells are making that protein, and then you're reacting against that protein. So as opposed to, some of the live virus vaccines like the live polio or the chickenpox or anything, it's not a whole virus. It's just one protein of the virus. It's actually an mRNA in most cases of the virus, so not even the DNA. So that coding sequence for that COVID protein is being expressed, and you're making an immune response to it. So it depends on which vector they're using to put it in there. So if they're using an AAV, which there are some that are being developed that way, that could be a problem for anyone trying to get AAV gene therapy. But most of the large ones are not using that. I mentioned the Oxford trial, which I think is a chimpanzee adenovirus. And again, adenovirus is not adeno-associated virus. The other companies, Pfizer and some of the others, are not using AAV to get that in. And so those shouldn't be a problem for those.
Great, thank you. I think that's going to be really helpful for those families. So we are going to move on to our next presenter. And so joining us today, we have Dr. Louise Rodino-Klapek, who from Sarepta Therapeutics. So Dr. Klapek, it's all yours.
Thank you. Thank you for inviting me to speak tonight. It's a really privilege to be here to meet with you virtually for the 6-minute talk test, and we'll see how well I do. So if you go to the next slide, my presentation will contain forward-looking statements, and that just means that I may make statements that involve risks and uncertainties, many of which are beyond our control, and you can visit our website to read these in much more detail. Today, I'll be discussing our microdystrophin program. I'd like to remind you that microdystrophin is investigational, has not been FDA reviewed or approved. If we go to the next slide. Sarepta has deep experience and commitment to Duchenne with over a decade of experience. We have 14 programs in Duchenne in total, which includes 14 in RNA, 4 in gene therapy, and 2 in gene editing. Next slide. We are committed to advancing science to develop the best therapies, and to that end, we are partnering to combat key immunology challenges, as Dr. Crate mentioned. We recognize the challenges of reaching each and every patient, but we're committed to doing our best in this case. The immune system currently limits eligibility with the presence of antibodies in some individuals. And so as we saw, typically you were using the viral vector to deliver our payload, in this case microdystrophin gene, to produce microdystrophin protein into muscle, whether that's skeletal or cardiac muscle. But when the antibodies are present in some individuals, it could block the transduction or the entry of the capsid into the cell and would make this ineligible. So we're trying to combat this in multiple ways. So one, by eliminating antibodies in positive individuals. This can be done through drugs that can potentially inactivate the antibodies or remove them physically. We're also looking at ways to prevent the antibodies from forming in the first place, and that's given with the first dose of gene therapy. And finally, we're looking at novel strategies using novel AAV capsids or other ways of delivering using non-viral delivery that the immune system can't recognize for future treatment. So to summarize, we're currently conducting research with multiple partners and internally to determine how we could progress these strategies to the clinic. So next slide. So I'd like to call attention to our most recent data releases for both our microdystrophin program, otherwise known as SRP-9001, and a related form of muscular dystrophy, LGMD2E, or our SRP-9003 program. For 9001, we have 2 ongoing trials. The first trial, Trial 1, was an open-label study done in Duchenne muscular dystrophy boys between the ages of 4 to 7. At a dose of 2 × 10⁻⁴ vector genomes per kilogram. And we recently published our 1-year data from this trial, and I'll go into that in a bit more detail on the next slide. We also have a second trial, Trial 2, which is our ongoing placebo-controlled trial. We've enrolled 41 patients, and enrollment is complete. This is also at a dose of 2 × 10⁻⁴ vector genomes per kilogram in patients who are 4 to 7 years old. And data from this trial will be released when the study is complete. And so next, I'd just like to mention our LGMD2E trial results, which we've also presented recently. So to remind you, this trial is a open label study as well, with 2 dosing cohorts. 3 patients were enrolled at a dose of 5 × 10⁻⁴, and 3 more patients at a high dose of 2 × 10⁻⁴, which is equivalent to our microdystrophin dose. This has important implications because this trial also used the same vector, RH74, and the same promoter, which is, you'll remember, is our light switch, which turns on the expression of the protein inside muscle cells. In this trial, patients between the ages of 4 to 13 were enrolled, and also between the weight ranges between 18 and 57 kilograms. And this is important as we gain experience with both the RH74 vector in terms of treating patients that are both older and also heavier. So we've disclosed data on 1-year safety and functional data for cohort 1, for cohort 2, day 60 safety and expression data. So if you go to the next slide, I'll go into more detail on study 1 results. We've recently published these as well as JAMA Neurology with Dr. Rendell. All right, so in terms of safety, we have seen no SAEs to date in this first study. 3 subjects had elevated liver enzymes, which resolved with steroid treatment within 1 week. And the most common adverse event that was treatment-related was vomiting, which occurred in 50% of patients shortly after AAV administration. There were no adverse immune responses or clinical sequelae associated with complement activation and no other clinically significant laboratory findings. You can advance. We performed biopsies at both baseline and at 12 weeks. And so the first question we asked is how much of the transgene got inside muscle cells, and we found a mean of 10 to the 5th vector copies per microgram of DNA detected. And you can advance to the next. We also looked at the total amount of protein made, and this is achieved by looking at Western blot. And we see 74.3% compared to normal. If we normalize for muscle content, it's 95.8% as compared to normal. Next, if you advance, we look at microdystrophin expression at the membrane. And we do this and quantify this in multiple ways. One way is counting the number of fibers, where we see 81.2% of muscle fibers expressing. And we also look at intensity, where we see 96% intensity compared to normal. And finally, if you'll advance to the following, we looked at functional outcome measures. Here I'm showing North Star Ambulatory Assessment. This is a compilation of 17 different functional measures. You can see at baseline, we saw a mean of 20.5 points on the scale across the 4 patients, whereas at 1 year, we see a mean of 26. This is a 5.5 increase from baseline, which is equivalent to about a 27% improvement from baseline, which wouldn't be predicted by natural history in this age group. These preliminary outcomes need to be confirmed. This is an open label study, and we'll confirm these in our placebo-controlled trial that's ongoing, or study 2. So if you advance to the next slide. So where are we in our development? As I mentioned, study 1 and 2 are on. We're continuing to follow patients in study 1, our open label study, and our trial 2, 41 patients placebo-controlled. All patients have been dosed, and we are dosing patients that received placebo first. Their crossover patients are now being dosed, and we'll release those data when the trial is complete. Finally, we'll be beginning our late-stage studies in the second half of 2020. This is a group of studies referred to Study 3, with the goal of expanding eligibility criteria across both mutations and ages. The first of these trials, of the trial 3 trials, will be in the same age criteria similar to trials 1 and 2. And we are also currently planning a trial which will involve individuals who are non-ambulatory. These studies will include commercial process material and will support global regulatory filings and access and reimbursement. So if you go to the last slide. So thank you so much for having me speak today. This 6-minute talk was difficult, I'll tell you, but I appreciate it. And if you have any questions, you can always reach us at advocacy@sarepta.com. So thank you, and Eric, I'll turn it back to you.
Thank you, Dr. Klepek. And we have a question for you. So excited to hear, the news of some of the partnerships going on. Can you give us maybe any timeline for when we might expect to see some of those in the clinic? I know there's always a lot of excitement and interest about how we can get around pre-existing antibodies and tackling challenges like redosing.
Sure. Yes, we're definitely working as fast as we can with those partnerships. We're performing non-clinical studies right now, research studies, to be able to prove that they're effective at either removing antibodies or preventing those from forming. And so we're in early stages, but we're moving as fast as we can. So.
Great. Thank you. And we're going to go to our next speaker now, which is Dr. Beth Belluccio from Pfizer. So if you're ready, Dr. Belluccio, I'll pass it over to you.
Great. Thanks, Eric. Thanks so much. It's really a thrill to be part of this phenomenal conference that PPMD is putting on, and really grateful to be included in this panel on gene therapy. So I'm Beth Beluccio. I'm the Global Clinical Lead for Rare Neurological Disorders at Pfizer. And if you go to the next slide, I have a very similar slide to Dr. Rudina Claypack about forward-looking statements. So then we can go on to the next slide, where I'm really pleased to report about our ongoing phase 1b study. We continue to enroll study participants, and in fact, this slide is already a little bit out of date. We now have dosed 15 boys, 3 at the low dose and 12 at the high dose, and we'll continue to enroll additional participants. Just to update everybody a little bit on the status with regard to safety, we have had a total of 3 serious adverse events. 2 of those Michael Binks presented about at the PPMD conference last year, and then the third he presented at a conference earlier this year in May, and we've had none since that time. At that conference in May, Michael Binks also provided a thorough review of our preliminary signs of efficacy that we think are very encouraging, and that full presentation is available at Pfizer.com. So I'm not going to review all those results here, but the main thing that's really important about the results of our phase 1b study is that they're supportive of us moving into our phase 3 that we're really excited about. So on the next slide, I want to talk a little bit about our CIFRIO phase 3 study, which is a randomized, double-blind, placebo-controlled study, as I think most people know. It is currently listed on clinicaltrials.gov, and so all the details of the study design are listed there. I've already also given a couple of presentations recently to describe some of those details of the study design, and those are available on the internet, including one that's a YouTube video, so it's kind of easy to find. But just to remind people, it is a global multi-center trial, and we are actively moving forward to start the study as fast as we can. We haven't paused our program at all due to COVID, although as people know, I think there's a really tremendous strain on the healthcare system right now, and that is having some effect on our sites and on their readiness and on some other factors that we don't have any control over. But the good news is we're still on track to start the study later on this year, subject to regulatory approval. So as I mentioned, this is a global phase 3 study, and we have multiple sites, as described a little bit more on the next slide. In the United States, we have many, many sites planned, and as illustrated in this map, we have at least one site, usually many, within every region of the United States. And really our aim here is to make sure that patients and families who want to participate in the study won't have to travel very far to a site. Now, each site, as it's really ready to and open to enroll, will be posted on ct.gov. So that's the place to look for sites that are opening, and one should be near everybody who's interested in participating. We also will have multiple sites outside of the U.S., and those will also be posted on the ct.gov site. Now, in addition to this phase 3 study, the CIFRIO study of in the ambulatory population, as I think many people know, we also are planning a global multi-center randomized control trial, phase 3 study of the non-ambulatory population. And before that study starts, we are planning to include a few non-ambulatory subjects in our ongoing phase 1 study. The numbers will be fairly small, so as investigators screen patients for enrollment, they'll probably choose those subjects mostly from their own clinics. But we're really looking forward to our much larger study that will have opportunities for lots of non-ambulatory patients. So now I kind of want to turn to a different topic on the next slide. And this is around seroconversion. That's kind of a big word, but really all it means is that if you go and you test for neutralizing antibodies and you're negative, that sometime in the future, a year or two later, if you've been exposed to AAV, you may have then developed neutralizing antibodies and you'll be positive. So going from negative to positive is what seroconversion means. And some people have been wondering whether that can happen if you've been around someone who's been treated with gene therapy. And this is sort of illustrated in the little cartoon on the right, where the boy in yellow is receiving his gene therapy. And then soon after that time, some of those AAV viral particles could be present in some of his bodily fluids, like his saliva. And then let's say his brother in the red shirt is nearby and comes into contact with his saliva, then that boy in the red shirt may develop neutralizing antibodies. We don't think that would make him sick in any way, but if he were someone who were interested in gene therapy in the future, then that might affect his chances. Now, again, we don't really know if this happens, but it's really important, I think, for us to understand if it happens or not. So at Pfizer, we're going to do a couple of things to try and find out. And if you go to the next slide, really a key part of our plan is something we're calling the household contact study. So this is a whole separate study, but it's sort of a sub-study to our phase 3 CIFRIO trial. In the sub-study, what we'll be doing is asking household members, family members of the CIFRIO participants if they'd be willing to volunteer. And what all they need to do is have 3 blood draws in their home, and we'll be testing their blood for neutralizing antibodies. And this way, when that study concludes, we'll be able to have actual information on whether or not people do have seroconversion when they're around somebody who's been treated with gene therapy. So I think this is really important for the entire community, and we're looking forward to being able to share that data. Now, until we get those data, we will be looking to take care of and help the siblings of boys who are treated. And the next slide describes a couple things we'll be doing along those lines. The most important thing is, in our CIFRIO study, if there are 2 boys who both qualify for the study, they meet the inclusion-exclusion criteria, we'll be randomizing them to the same cohort. And that eliminates the chance that one boy could get the investigational gene therapy and then cause the other to develop antibodies before he can get treated. That really wraps up what I wanted to talk about today. If you go to the last slide, I just wanted to really express our gratitude to all of our partners who have given us a lot of input on our trial design by letting us know what's really important to patients, and that really helps us be able to bring therapies that are important to this community forward. So thanks a lot. Back over to you, Eric.
Thank you, Dr. Belluccio. I think we're all going to be really excited to see the results of that seroconversion study that's asking a really important question that we need to address. I was wondering, though, if you could expand a little bit on, in the slides that mentioned, in response to the serious adverse events that you had previously seen, there were some additional safety measures. Could you maybe expand on what you are doing to help mitigate those risks?
Absolutely. So really the most important thing we've done is to learn how to anticipate what's going on with these patients. So there seems to be a pretty set pattern of the response, and we can, and therefore observe it and monitor for it very carefully. So we have patients stay near the site for a couple of weeks. We can monitor their labs very closely, and that way we can really be on top of anything that could be happening and treat it effectively. And we're really hopeful that is going to be good in the future.
Thank you. And with that, we are going to go on to our next presenter, Dr. Fulvio Mavilio from Audentes Therapeutics. So I'm going to pass it to you, Dr. Mavilio.
Good evening, everyone. And thank you for inviting me to this extremely interesting symposium. In the next slide, this is the usual disclaimer slide. I think we can skip it in the interest of time and go to my real first slide. Audentes is a biotechnology company, the mission of which is to develop genetic medicines for devastating diseases with the potential to be transformative, and particularly for neuromuscular diseases. Audentes was recently acquired by Astellas, a multinational pharmaceutical company. So now we are a division of Astellas and a so-called center of excellence for genetic medicines. The next slide is our pipeline. As I said, we are focused on neuromuscular diseases. We have in advanced clinical development a gene therapy, an AAV gene therapy for X-linked myotubular myopathy, a second gene therapy product for Pompe disease, which is at the beginning of its clinical phase, and the first 3 products for Duchenne muscular dystrophy. I will get into more details of this, but these are gene therapy products aimed at skipping exons, and in particular exon 2 or exon 55 and exon 53, and finally another product for myotonic dystrophy. So in the next slide, what we are doing for Duchenne. At this point, after yesterday's session and after great Dr. Krebs' introduction, you know everything about AAV and you know everything about exon skipping. So what is that we are doing? We are essentially using exon skipping, which is pretty much what the antisense oligonucleotides that are developed by several companies, and you know about this, are doing to skip exons to restore reading frames in many mutations of Duchenne muscular dystrophy and allow for the production of a functional dystrophin product. So we are doing exon skipping, but what is different is the modality of delivering this interesting molecules. What we are using is a gene therapy vector, is an AAV that carries a gene like all AAV vectors, but this gene doesn't produce a protein like the microdystrophin you've just heard about, but rather a small nuclear RNA, which is the hairpin structure that you see in the right part of the cartoon, that carries this little red bar, which is an antisense sequence, very similar to what you know in, for the antisense oligonucleotides, except that this is not an oligonucleotide. It's an RNA sequence which is part of a larger RNA, which is the structure you see in this cartoon. So essentially, we are using antisense to skip exon and reconstitute reading frames, but with a different modality that we believe is more efficacious. Why? First, because we use a vector that puts this molecule, this effector molecule, into the majority of fibers in the muscle and in the heart, which is very important. Second, because this antisense is an RNA molecule and the splicing machinery normally into cells is based on RNAs and not on DNAs. Third, because this small nuclear RNA, this hairpin structure you see here, is actually a modified RNA, which is normally part of the splicing machinery. So we are actually targeting the antisense in the nucleus of the cells where the action is and when we think the skipping is going to be more efficient. Now, preclinical models in animal models of DMD with that mimic mutations that are typical of patients, we have observed excellent delivery of these molecules to the majority of muscle fibers and to the majority of the heart fibers. We have seen exon skipping up to 80% of the RNA transcript that comes from the dystrophin gene and robust synthesis of functional dystrophin. So based on this type of preclinical studies, in the next slide, the team of, before going to that, what's the result of this skipping? Well, you know the game at this point. Dystrophin is a huge molecule, and all those colored boxes give you a sense of how many domains, functional domains, are present in the dystrophin. So normal human dystrophin has this different domains of different colors, an amino terminal in red and a carboxy terminal in purple, and other domains in the middle. In making microdystrophin, we have to remove some of these domains, so have a protein that is missing some of its important function. Exon skipping, no matter what modality you use, essentially gives rise to a larger molecule, which carries an internal deletion that depends on the patient's mutation, of course, but it's essentially much larger than a microdystrophin and contains most of its functional domain. In particular, the carboxy terminus is very important, the NOS, so-called binding domain, which is also very important, and of course, the length, which is important for the mechanical properties of the molecule. So as I said, based on preclinical studies done with these modalities of exon skipping, in the next slide, the team of Kevin Flanagan and Megan Waldrop is the principal investigators of the study, is bringing this to the clinics. So this is a first in human. It's a proof of concept study for patients that carry a duplication of exon 2. So the skipping here is removing one or both of these duplicated exons. 2 patients have been already treated in this trial with a starting dose of 3 times 10 to the 13 vector genome per kilogram. The patients have been dosed right before the COVID-19 crisis, but unfortunately, COVID-19 delayed a little bit the analysis of these patients at the first time point. And the Megan and Kevin will report on the result of the first biopsies as soon as they are ready to do that. This study is done in collaboration with Audentes, with collaborators with this. We hope to capitalize from the knowledge that this pilot study will give us in terms of efficiency of skipping, efficacy of producing dystrophin, and possibly ameliorating functional parameters in patients. And based on that study, we will fine-tune the development and construction of our vectors that, in the last slide, are essentially aimed at skipping of many different exons. Here in this pie, you essentially see the proportion of patients that carry mutation that can be corrected by skipping one or the other exons. For instance, 13% of patients are amenable for exon 51 skipping, 8% for exon 53 skipping, and so on and so forth. So with 6 to 8 products, we believe we can address more than 50% of the prevalent patient population. And of course, developing 5 or 8 different products is 5 or 8 more times more difficult than developing one. So the way we want to do this, and we may want to we can address this in the discussion if we have time, is through a platform approach. So essentially developing a series of vectors that would be absolutely identical in everything except for the small antisense sequence, which will, of course, will be specific for each mutation. This work is ongoing, and as I said, essentially for duplication of exon 2 and for exon 51 and 53, which are of course, the most frequent prevalent population in humans. And with that, I will stop by thanking you for your attention and the PPMD for giving us the opportunity to discuss this, that as you can see is at the very beginning, this journey into gene therapy, exon skipping gene therapy. Thank you. Back to you, Eric.
Thank you, Dr. Mavilio. We're always excited to see new strategies enter the Duchenne space. Now, we do know that many in the DMD community have heard about the 2 deaths that occurred in the X-linked myotubular myopathy gene therapy trial. We were hoping you could share with the DMD community a summary of that situation and what is known at this time.
Sure. Well, first of all, we're very saddened to share the news that 2 patients unfortunately passed away in our MTM gene therapy trial. And of course, our thought as a company and as scientists and members of the rare disease community go to these families in a very, very difficult moment. So what happened? As I said, 2 patients passed away. And of course, we are committed to be as transparent as possible and give the patients community, not only the MTM patient community, all the information that are necessary to understand what happens. First of all, we have treated 23 patients in the MTM gene therapy trial. 6 patients at the lower dose of 10 to the 14 vector genome per kilogram, and the rest at the higher dose of 3 times 10 to the 14 vector genome per kilogram. The patients that unfortunately died were part of this second cohort of patients, so they were treated at the higher dose. But that's not the whole story. So the immediate cause of death for both patients was sepsis, which means generalized infections that could not be, unfortunately, controlled and eventually caused their death. But this came at the end of a pretty long period of hospitalization due to liver conditions, to what became severe liver dysfunction, and actually started 4 weeks after dosing as elevated bilirubin levels. This is something that we had not observed before. As it turned out, a significant fraction of the MTM patients do carry a liver condition that is a mild fluctuating bilirubin levels, and this appears to be part of the disease. It was probably overlooked in previous natural history studies, but in the natural history study that we carried out before and in parallel to the interventional studies, we did observe that a significant number of patients carry these conditions. So these patients were, the 2 patients that died were older than the average patients in the trial. They were more than 5 years of age. They were heavier. And since the dose is adjusted to weight, as you know, they also received the highest total dose of vectors. So at the moment, of course, the investigation is ongoing. We don't have any definitive conclusion to propose to you. But what we believe is that it is a combination of factors, but the pre-existing liver fragility we can define is probably an important component of this for a vector like the AAV that massively goes to the liver, that could induce liver toxicity that is in general well tolerated or controlled. In these patients could be in fact devastating. We are discussing with the agency, of course, trying to introduce corrective measures in our therapy in order to reduce the risk for these patients. I have to say that other patients that were younger and smaller with similar pre-existing liver conditions that were treated at the lower dose did not develop any serious adverse event. So we are learning and we are working with the agency in order to reduce the risk for this patient. I just want to conclude by saying that MTM and Duchenne are very different diseases. MTM is a devastating, lethal disorders in the first 2 years of life. So what we are seeing here and what we are learning is something that would be important for us to learn also for our future gene therapy trial for Duchenne. But you have to consider that the risk for these 2 populations is very, very different.
Thank you. We know that's a really difficult subject to discuss, but we do appreciate the transparency, and I think everyone in the Duchenne community appreciates that as well. And so we're going to move to our next speaker. We have Dr. Joel Schneider from Solid joining us. So, Dr. Schneider.
Thank you, Eric. Good evening. My name is Joel Schneider, and I'm the Chief Technology Officer at Solid Biosciences. I'd like to thank PPMD for hosting the event this evening. And tonight I'd like to give an update on our Ignite DMD clinical trial, testing a microdystrophin, as well as a bit of an update on a press release, which we put out immediately before the panel this evening. Next slide, please. These are my forward-looking statements, like the others. Next slide, please. As a reminder, Solid Biosciences was founded in 2013 by families directly affected by Duchenne muscular dystrophy. Since then, we've been developing different therapies for Duchenne, including our microdystrophin gene therapy candidate called SGT-001. As others have described, the candidate is designed to replace the underlying deficit associated with Duchenne, which is the absence of a protein called dystrophin. Next slide, please. SGT-001 contains a differentiated microdystrophin construct, and it's based on 30 years of research by our advisors, Dongsheng Zhuan and Jeff Chamberlain, and it shows robust protein expression as well as the unique restoration of a protein called nNOS, which Fulvio mentioned before. The image on the right highlights data from one of our clinical biopsies taken 3 months after administration in our 2 × 10^14 vector genomes per kilogram cohort, showing the expression of a microdystrophin alongside the restoration of functional nitric oxide production in the muscle bed. Next slide, please. Ignite DMD is our phase 1/2 clinical trial, which is currently on hold. And to date, we've dosed 6 patients in the trial, 3 at the dose of 5 × 10^13 vector genomes per kilogram, and 3 at the dose of 2 × 10^14 vector genomes per kilogram. We have 2 primary endpoints, safety, of course, as well as SGT-001 microdystrophin expression 12 months after administration. We also have a number of secondary endpoints associated with the trial that allow us to better understand efficacy and functionality of our microdystrophins. As a reminder, SGT-001 and Ignite DMD are currently on hold because of observations of serious adverse events in the last patient dosed. And I'll talk a little bit more about that in just a moment. But importantly, as a reminder, all patients within the trial are doing well, including the last patient dosed. Next slide, please. We're encouraged by the expression that we've seen to date. And SGT-001 dosed at the 2 × 10^14 vector genomes per kilogram dose leads to robust, widespread expression across the muscle bed, as highlighted on this slide. Next slide, please. So where we are today, we have, as I highlighted in the press release that came out just before this gene therapy panel, we heard back from the FDA today that the clinical hold on Ignite DMD will continue in place. And they specifically responded with requests for further manufacturing information related to process improvements that we're making in the SGT-001 production process. They've also requested updated safety and efficacy data for all the patients dosed, and they provided additional directional information on the total viral load to be administered per patient. Our clear next steps and immediate goals are to engage with the FDA to determine the path forward for the program. And we're encouraged by the microdystrophin expression that we've demonstrated to date. We continue to follow the current study subjects per protocol, and we're looking forward to evaluating samples and functional endpoints taken from 12-month time points. These, of course, are being done with consideration of the current COVID-19 environment. I understand that there are many questions related to our hold and our next steps, and we'll share updates regarding Ignite DMD with the community in the coming months. Next slide, please. We're grateful to the community for your continued support and the interest in our program. And together, we're going to continue in our relentless pursuit to improve the lives of patients with DMD. Our sincerest thanks for PPMD for quickly adapting this trial, even regardless of the COVID-19 pandemic, and for giving Solid the opportunity to be part of this really intriguing panel. Thank you for your time, and back to you, Eric.
Thank you, Dr. Schneider. And thank you as well for sharing the comments that you received already from the FDA. I know that's something as pretty quick turnaround time, but again, we appreciate the transparency. I actually wanted to follow up on something from your construct. We have heard about the nNOS binding domain before. Could you maybe clarify for the community what you see as the significance of that, and just kind of dive into that a little bit?
Sure. As Fulvio highlighted, normal dystrophin expression is correlated to the expression of a number of dystrophin-associated proteins that play various roles in normal muscle function. Nitric oxide is produced in a healthy muscle during periods of energetic need. And so when we exercise and the muscles require more oxygenation, nitric oxide is produced, oxygen is supplied to the muscle beds, and that allows us to respond to energetic needs. Duchenne patients lack dystrophin and therefore lack the restoration and localization of these other critically associated proteins, and therefore lack nitric oxide. And in preclinical models, we've been able to demonstrate that the restoration of microdystrophin alongside the association and restoration of normal nitric oxide production leads to various differentiated functional improvements. We were excited to see in our patients dosed with SGT-001 in the 2E14 cohort not only the expression of microdystrophin across the muscle bed, but also functional nitric oxide production in the muscle as well. We also saw the localization and expression of the nitric oxide synthase protein as well. Now, we've got a lot more in front of us, and today we're really focused on removing the hold and working with the FDA as closely as we can. We are excited by the data we've seen so far and the unique differentiation in our microdystrophin construct.
Thank you. And we're going to open it up now to our full panel for a Q&A. So again, if you have questions, please submit them below. I think we are going to just kind of cancel our last polling for tonight. We have a lot of questions. We're not going to be able to get to all of them tonight, but we will pass those along to the sponsors. And just as a first one, Dr. Kripe, I'm actually going to throw this to you. We've had a lot of questions come in now that we see different AAV serotypes, RH74, AAV9, AAV8, and a lot of patients and family members are asking, if I test and have pre-existing antibodies to one of these vectors, is it possible that I could still receive gene therapy from another one?
Oh, that's a great question. A lot of the antibodies do cross-react. So even technically, the word serotype originally meant that you only reacted to that type and not to other types. A lot of the AAVs do share a lot of common sequences or epitopes, which is the term we use for what an antibody recognizes. And so many of the antibodies that you might generate for one virus will cross-react to other AAVs. So the only way to know is to get the eligibility testing for any given study where they test for antibodies for their particular vector.
Thank you. I think, Ryan, you had a question?
Yeah, so, and this is really for all, and we'll probably just start around the circle. These are early days, and families are having to make decisions. And I guess the big question for so many is the uncertainty around durability. So you're making a choice for a gene therapy, and it's a one shot for now. We're all talking about redosing. I think we can admit it's not around the corner. So I just wanted your general thoughts on this sort of first generation for Duchenne gene therapies about durability based on preclinical and even based on the early results we're seeing. So, Joel, I'm going to pick on you because you are on the top right of the square there.
It's a little like the Brady Bunch.
It totally is.
It's a great question, Ryan. And enrolling in a gene therapy clinical trial is a personal and difficult decision that, of course, should be done in consultation with your physician. It is early. This field is very nascent. But I think in general, the data that's been generated so far is certainly promising and encouraging. The best data that we've generated from our preclinical models is in our dog work, our dystrophic dog studies, where we've gone out for biopsies serially taken over various points of time and seen over 3 years of expression. Now, a dog isn't a human, and a mouse isn't a dog, and a mouse certainly isn't a human. And so we have a lot more to understand as we all assess durability. Louise highlighted their 1-year follow-up, which is, of course, also very encouraging. And muscle does seem to be uniquely suited for maintaining transgene, which is the gene that we're delivering, maintaining durability alongside the expression. And it may be because muscle tends to be a fairly slow tissue to turn over.
Okay. Louise?
I would agree. And, durability is a tough thing to assess because there's no way to speed up time, right? So we continue to gain this information over time and relying on non-clinical models and clinical data. So we're encouraged by the clinical data that we have so far in our microdystrophin program up to 1 year, and then also in the limb-girdle program as well, where we didn't see any drop in function over that time. We saw sustained functional improvement. And so together with non-clinical data, we have mouse data for the lifespan of the mouse. There's data from the golden retriever muscular dystrophy dog for at least 6 years. So I think as the field's collectively collecting this data, I think we're encouraged about the durability of treatment, but there'll be many years of follow-up and continued study to be able to anticipate the total outcome.
And Beth, I see you shaking your head, and imagine you you agree with those statements. Anything to add to that?
No, I mean, I think we have sort of similar data that aligns with what people have said. I think the only other thing is that really the most important thing about durability is how patients will do, right? How they'll function. And so I think all of us probably have trials that are going out for a prolonged period. I know our trials are all at least 5 years long, so that over time we will be able to track, how patients really function and whether that's maintained over the long period.
Yeah. Fulvio?
I can give you two pieces of data. The first is for myotubular myopathy, we carried out for years studies in the dog model. The earliest dogs that were dosed are now 9 years of age, and they're still doing fine. Actually, they became the pet dogs of the investigators. So in a model which is not a dystrophy, so it's a different disease, which like myotubular myopathy, at least in the dogs, we have a duration that can go up to 9, possibly 10 years. The second piece of evidence comes from studies that I carried out in my previous life as an academic investigator on the dog model of Duchenne. And what we found is that what is very important in order to durability to be long is the number of fibers that you can actually transduce with your vector. What we found out is that if we transduce, or if we put our product that in those studies was microdystrophin in less than 50% of the fibers, regeneration, degeneration will continue to go on. And then you will essentially lose your vector much more quickly than if you transduce more than 50% of the fibers. And if you transduce more than 80% of the fibers, that stabilizes the degeneration regeneration cycle and essentially grants a much more durable effect. I think that this is important, and I was very pleased to learn from your presentations that in all these clinical trials, the number of fibers that are transduced with microdystrophin are actually very high. That's an important issue, and it's important when we think about dose escalating. It's difficult to start with doses that we can predict are not going to allow for a durable effect. And that's part of the reason why we all tend to start with doses that are relatively high.
Thank you. Just to follow on to that, as we get into the older non-ambulatory patients with less healthy muscle tissue, the risk certainly may be going up then if these are high doses of virus. I hate to kick it around the room again, but this is a population, we're really looking at, we want more treatments for, but what does that look like in terms of the risk benefit as patients get older and there's less healthy muscle to essentially play with, for lack of a better phrase, or treat? We're going to go around the room again, and we're going to start with Joel.
Sure. Thanks, Ryan. I think moving forward in general in this field, we're taking the iterative, stepwise, cautionary steps with the ambition of ultimately, hopefully, dosing all patients with Duchenne. Again, from the preclinical world, there's data that's been generated in older animal models that suggests even with decreased amount of muscle mass and increased inflammation and fibrotic presence, certainly animal models can, of course, still benefit. Solid's first patient dose was a non-ambulant patient. And so our ambition is certainly to go back there. Today, of course, as I highlighted, we have to work to have the hold lifted. But I think the overall approach that you're hearing about is really humbled in the sense that we're all taking safe, cautionary steps forward, trying to understand the body's response to these major infusions of viral particles, and based on the knowledge that we're generating in the clinical experience, making the responsible steps forward.
Mm-hmm. Yeah, and in the interest of time, unless somebody has a strong disagreement, I want to let Eric ask another question, or if someone wanted to add anything to that, please do.
All right, great. So we've had a number of questions come in from different families asking about specific mutations, whether it be duplications, nonsense mutations. And again, we're going to go around to everyone, and maybe you could expand on, in either your current trial or upcoming trials, any restrictions that you have around specific mutations. And Fulvio, I'll actually start with you on this one.
Well, of course, exon skipping is by definition mutation specific. So for each one of our product, we will enroll patients that have mutations that can be corrected by skipping one particular exon. So again, by definition, the type of product that we are developing are not addressing the whole patient population, are actually addressing a fraction of the population for each specific product. We hope to be able, through the platform approach, to have products that will present the same toxicity, the same risk, the same efficacy in terms of delivery and exon skipping and target each specific mutation. But clearly, with our approach, we cannot possibly address all the mutations.
Great, thank you. Beth?
Right, for our AAV gene therapy, exclusion at all for underlying mutation.
I'm sorry, could you repeat that? It was a little bit choppy coming through.
Oh, so we're having a little trouble, so let's move to, well, actually, you're back, Beth?
Yeah.
Yeah, there you go. Yeah, you're back. Go for it.
Yeah, okay, good. Short answer, we don't have any exclusions for underlying mutation.
Right.
In terms of mutation, you just have to have one that corresponds to Duchenne.
Thank you. Louise?
So our first two trials, we did have an exclusion for mutations between 18 and 58 of the dystrophin gene, but for our future trials, we will not have those same restrictions on mutations.
Great. And Joel?
We have, can you hear me?
Yes.
We have no exclusion criteria related to underlying mutations, just a diagnosis of DMD.
Great, thank you. This one came in for Audentes. Do you have a timeline for when you'll have your IND submission for your phase 1 trial?
We are developing our timeline. As I said, we are essentially collaborating with Nationwide on a first proof of concept trial. Our timeline in terms of Audentes specific products will largely depend on the preclinical studies, the discussions that we're having with the agency, the result of the proof of concept studies in terms of dose, efficacy, and toxicity, of course, and safety. We don't have a real timeline yet, but we are confident that
Thank you.
I heard he's confident.
I think that might have been on our end. So we have another kind of broad question that we've had come in a number of different times, and it's questions about steroids, both leading into the study and kind of coming out after receiving gene therapy. So knowing that different families are on weekend dosing versus daily dosing, prednisone, deflazacort, are there any restrictions or that for families as they're looking to join trials that they should be considering, either kind of preparing for a trial or kind of after the delivery phase? And so I'll start with you, Louise.
Sure. So if our current trial, patients are on daily steroids for the first 30 to 60 days for delivery. For our next trials, we'll continue a similar paradigm, and we'll, as we release the design of our next trial on clinicaltrials.gov, we'll sort of go into a lot more detail on the specifics of our future protocols for the next studies.
Thank you. Beth, could you share what is planned for the CIFRO study?
Yeah, absolutely. So our patients need to be on a daily dose of steroids coming into the trial. We have specific minimums that it's hard to remember them if I just say them out loud, but they are listed on our ct.gov site in terms of the minimum doses, because it's a little different for prednisone versus deflazacort. We do then increase the steroids slightly right after dosing, and we found that to be very effective in minimizing some of those immune responses. And then the patients taper back down to their background, their dose after that.
Great, thank you. And Joel, you as well.
Very similar design to Pfizer, where for 6 months before administration, patients need to maintain a steady course of their standard of care. The dose is slightly elevated before administration, and then subsequent to administration is tapered back down to their standard of care over a period of time.
Great, thank you. And I just want to take this time to thank all of the participants today in our gene therapy panel. We really appreciate you taking the time to be with us today. Dr. Kripe, again, thank you for that wonderful introduction. But we had a lot of questions that we weren't able to get to. We do apologize for that. We will make sure that we get those to the sponsors. But we wanted to, again, thank you all and talk about what's going to be going on over tomorrow.
Thank you, everybody.
Thank you.
Okay. So that brings us to the official close of day 3 of the virtual conference. Again, thank the panelists from today, all the presenters, did a fabulous job. Tomorrow's our busiest day. It's our longest day. You'll get an email in the morning with a schedule. And Eric, what is ahead of the day tomorrow?
So yeah, we have a lot going on. To start off the day, there is going to be a yoga session for parents, so you can get a little loose and limber before we get started for a long day. We are going to have a session talking about caring for female carriers. We know it's a really important topic, and we want to make sure that all of our carriers are taken care of. We're going to have a fantastic keynote address from Rachel Callender about how language shapes our landscape. And we're also going to take another look at our In the Pipeline series. We're going to talk about some of those therapies that are combating fibrosis and inflammation. We're also going to have some more care panels. We're going to discuss the role of physical medicine and rehabilitation. And we're also going to explore why cardiac intervention is so critical in Duchenne. As we always like to say, the heart is a muscle too, and it's one we want to make sure we're looking after. And finally, we are going to have a look ahead at some of the emerging strategies in Duchenne.
Great. So teens and adults, you will have a specific track on quarterbacking your own care. So be on the lookout for information on that session. And then to end the virtual conference, we are going to have a conversation with a good friend of ours, Joel Wood from the Foundation to Eradicate Duchenne. FED, as they're also known, has been a longtime partner with PPMD in our advocacy efforts. So it'll be great to have Joel help us end the conference. So check your email tomorrow for the overview. With that, we wish you a really good night, and we will see you tomorrow.